sound is turned down way low. Hold on a second. All right, that works. All righty. Let's go ahead and get this show on the road. So for the first time, we've got the whole rest of our schedule on the board. So we are rapidly approaching the end of this. We are into our 5th section of this class, our last organ system. Although really it's kind of like 2 organ systems that are obviously closely related to each other, as we'll be talking about the reproductive system. For the first time, we'll be talking about significant differences between the male and female anatomy and function of this. And so we'll start by talking about some of the basic stuff today, and then we'll move on to the reproductive anatomy. Thursday is a holiday, so eat turkey with just you and the closest people in your family. Don't be one of the 3 million who have traveled. If you have already, well then good for you. I was looking at the schedule. Tuesday, we don't have anything scheduled, and it's the first day back after the holiday. So I'll be honest, I haven't planned a daily quiz yet, but that does look like a good day for one. So maybe we'll do that. I don't know. We'll see. You should be thinking about such things. But we do still have definitely 3 more assignments that are going to be due. You've got your Unit 27 review that's going to be due, and then 2 labsters that are necessary. I know one of them is called endocrinology, but if you look at it as you do the activity, you'll see why it's much more relevant for the reproductive system. That we're doing that. And again, remember, you need to complete the whole thing and get 80% correct for full credit. Remember, because it is linked through Canvas, when you complete it, if you get 98%, you get 98% of the points. But remember, after the assignment is completed and after the due date, I go back in and I reset to the grades to full credit. So it's automatically input. So don't send me an email, hey, I got 85% on it, but it didn't give me my full 20 points. I'm the one who has to give you the full 20 points. So and I'm not going to make those changes until after they're all due. And then Thursday, the 10th, we have our lab and lecture exam. Same as ever was. You know what to expect from that. And then the following Tuesday, so you have 5 full days to study for, we have our final exam. Again, the final exam is cumulative, covering everything we have talked about in this class up to this point of time. And there are 3 important things to remember. The first is it does include lab and lecture material. However, it is 100 multiple choice questions, right? The really, really good thing about multiple choice questions is that the correct answer is there. So you don't have to pull the information out of the ether. You just have to recognize the correct answer. And that is a very, very different skill. The downside of that is, as you know, multiple choice questions almost by definition are tricky. They have that one word in there that if you misread it or if you misinterpret the phrase, they've got that trap answer there to catch you. So you have to read them correctly so you can answer them correctly. Read them carefully so you can answer them correctly. I will say that the questions are all new questions. None of them should be questions that you've had on previous exams. I've always felt that previous exams is a worthless way of studying for the final exam. Again, what you have to remember is that those previous exams only cover a small portion of the material that you are responsible for in this class. It's impossible to put everything on an exam. And so just because something was or was not on the lecture exam doesn't have any influence on whether or not it would be on the final exam. Again, I don't reuse questions. It's a completely different test bank. So again, be ready and be prepared for that. It is still multiple choice questions, so that you should be expected. The only thing that would be different that you need to be aware of, the only different types of multiple choice questions is because it does involve some lab material. There will be images, pictures of models or materials or illustrations or something like that with certain things labeled on it, and you may have to identify the thing that is labeled. Because obviously that would be something that would normally be in a lab exam. The other thing, lecture exams are processes. So normally if you have to describe a process that has 5 steps, you have to describe out those 5 steps. So instead what you may see is multiple choice questions that have the 5 steps of a process and you have to put them in order. right? You have 4 different options, 5, and then 2, then 4, then 3, then 1, and like that kind of thing. So other than that, I mean, it's the same kind of questions you've seen before. I think those would really be the only things that would be different from that. All right. Questions on any of that? Excellent. Oh, either my microphone's not on, although my good microphone's not plugged in. So let's plug that in. Hopefully you guys at least heard the first part of what I said, so because I don't have to repeat all of that again. For the final, it will be 100 multiple choice questions. And again, this test counts as a test. This is 100 points towards your total points in the class. However, remember also that to reward you for gaining and retaining this information, I will allow you to replace your lowest lecture score with your final score. If it helps you, of course. If your lowest lecture score is an 82 and you get a 79 on the final, I'm not going to penalize you by replacing that 82 with a 79. But if maybe you took a less vigorous 430 instructor and you got a 40 on the first lecture exam and you get an 85 on the final exam, then you get 45 more points dumped into your bucket. So for some people, this can make a pretty good swing. But again, regardless of whether you use it to replace a lecture or not, it does count as a grade on its own. Unless you're doing really, 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 really good in this class, you have to take the final. Right? I've had a student in the past who did really, really well, and he only needed like 54 on the final to maintain his A, and so he focused on the other classes he was doing, and that's fine. If you get enough points in your bucket, you can do that. But I don't think anybody in this class is doing well enough right now where they cannot take the final at all. Yeah, I'm pretty sure that's what I just said. It's going to be material from both the lab and the lecture and 100 multiple choice questions. So yes, those of you who had me for 430 last semester when we're online, you know exactly what that is like. All right. Any other questions?
How are last exams doing?
So you guys and my 430 class both had exams last week. So I just finished theirs yesterday. So I've just started on yours. It is slow going. I've just barely cracked into the first couple exams. So I really even haven't gotten an impression of them yet. I think I'm only 2 or 3 lab exams in. So my hope is, again, I've got class tomorrow as well. So again, we don't have big plans for Thanksgiving. So I figure by Thursday or Friday, I should have the exams graded and posted Friday, Saturday at the latest would be my guess. I'll post the grades, and then as I've done before, and again, especially because of being a holiday weekend, I will wait till Tuesday next week after lecture to release the exam so that you then have the opportunity to spend some time looking it over, seeing if there's any questions. And then you can always come to my office hours on Wednesday if you have questions about that. All right. Any other questions? All righty, excellent. Let's go ahead and get started then. So as I mentioned, our goal is to discuss the very last organ system that we have left, and that is the reproductive system. However, as we've also talked about, there are some unique characteristics to our reproductive system. For starters, there we go. It is really the only non-continuous functioning organ system. Right? Your cardiovascular system doesn't get to take every other Saturdays off, right? Your muscular system doesn't get holidays and 2 days in Christmas time or anything along those lines. Most of our organ systems that we've talked about up to this point of time are pretty continuously functioning. And even our immune response, which again is not an organ system, it fluctuates in its functionality, starting a little bit low and then building and slowly declining over time. But certainly the endocrine system, the lymphatic system, those systems around them are fully functioning. The reproductive system is the only non-continuous functioning system. Right? And the other big difference between the reproductive and the others is, as I mentioned, there are significant, and again, we've talked about many differences between males and females, size, volumes, all sorts of things along those lines. But as we've also talked about, none of those are really meaningful. This is the first time we're really going to talk about any kind of meaningful differences, sex differences between males and females. Now, let's start easy. What are the primary sex organs? Right, these are the easy questions, and these are the points where I expect you to respond. Excellent. Exactly, right? This is indeed an anatomy and physiology class. We are allowed to say penis. We are allowed to say vagina, vulva, gonads, all of those things, absolutely. But penis and vagina, while incredibly fun to say and useful parts of the reproductive system, they're really just the copulatory organs. The primary organs, indeed, generically, are what are known as the gonads, and those gonads indeed involve the testes in our males and the ovaries in our females. Yes, the vagina and the penis are organs in the reproductive system, but as I mentioned, they're really just the copulatory organs. Our primary organ that serves the primary function is the gonads. And what is the primary function of the reproductive system?
Production of sperm and eggs.
Yeah, there you go. Okay, excellent. Our job is to make our gametes, again, a generic term, and the specific examples of those would be the ova and the spermatozoa. Absolutely. And what do we use those puzzle pieces to do? You put those two puzzle pieces together, and what do you get as a result? Yeah, other people. Exactly. You get other people as a result of it. Absolutely. So exactly. The function of our reproductive system and of those gonads is to produce the gametes, the sex cells. Again, hopefully you've done your Labster, so you're prepared to understand the things that we're going to need to talk about today, because we are going to get deep into vocabulary today, like we always do in anatomy and physiology. So we have a lot of that to talk about. So we make those gametes, or as we refer to them to my daughter when she was young, the puzzle pieces, and you put those puzzle pieces together to make a little baby. And again, the plural of those are spermatozoa and ova, right? What we commonly refer to as the sperm and egg, but the singular is spermatozoon and ovum. However, remember, let's not also forget that as part of the reproductive system, the females get the honor and privilege of housing those puzzle pieces, nurturing those puzzle pieces until they come the size of a basketball, and then expulsing that basketball out of their bodies. So they get to house and develop and nurture those new people until they're ready to be expulsed. And of course, not necessarily a part of the reproductive system, but definitely related to many of the reproductive functions, is that our gonads, the ovaries and the testes, produce the sex hormones. What are the three classes of hormones that are produced in the gametes again? I mean, in the gonads? Classes. Three classes of hormones produced by the gonads. Excellent. Androgens, and again, the most common specific one is testosterone. Estrogens, the most common one of which is estradiol. Remember, estradiol is the specific type of estrogen. And what's the third? Progestins, the most specific one is progesterone. Excellent. So we have those. And as we talked about in the endocrine system, those classes of hormones play a role in the growth and development of many of the organs and tissues in the body, as well as the maturation and development of the reproductive organs. Those sex hormones, the changes that they undergo in our body is what helps us to tell the difference between a boy and a man, or a girl and a woman. And we also talked about how they play a role in libido, they play a role in aggression, and they play a role in behaviors like that as well. All right. Now, our goal is to understand the reproductive system. just like in other classes on campus, the goal may be to understand how an engine works. And if you're not very familiar with engines and you wanted to understand how they worked, and you had two cars sitting in front of you, you had a 2021 Lexus and you had a 1968 Volkswagen Beetle, right? Both of these are cars, both of them have engines. So which one are you going to want to look at first? Does it matter if you're trying to understand how an engine works? Which one are you going to pick?
The simplest one.
Yeah, the Volkswagen, right? Absolutely. You're going to pick that Volkswagen. That Volkswagen Bug engine is tiny. It's simple. It does basically the same thing the one in the Lexus does, right? But you can pretty much pick up and hold it in your hand. It is a much simpler version. Doesn't have near the bells and whistles that the Lexus has. And that's exactly what we're going to do here. In understanding the reproductive system, we're going to start first with the bug. We are going to start with the simple version, the basic version, and then we'll get to the 2021 Lexus with all the extra bells and whistles, and having a basic understanding of how the engine works will help us to understand the female process. So our goal first will be to explore the male reproductive system. The male reproductive system is made up of those all-important gonads, the testes, and basically a closed duct system. What do I mean when I say that this is a closed duct system?
It's not exposed to the outer world.
Well, but is it exposed to the outside world? Is it all inside the body? What do the testes produce again?
Semen.
Semen. Well, they particularly produce the spermatozoa, the sperm portion of the semen. Absolutely, the spermatozoon. Absolutely. And it is produced here, right, in the testis. In the testis, when it is produced here in the testis, how many places does it have to go? One. Basically, it enters into all these tubes, and each of these tubes just has one route that it can take. It goes from the testis into all these different efferent ductules and the rete testis and the epididymis and the ductus deferens and all these things, and it basically has just one path it can take. So from the testis to the outside world, it only has one route that it can take. Now, why do I make a point of mentioning that here with the male? If I have to specify that the male is a closed duct system, being the smart, sophisticated student you are, what does that tell you?
Female is not.
That the female is not, absolutely. The female's duct system is an open duct system. It's one of the things that makes sexually transmitted diseases much more dangerous for females, because the female ductwork system is actually open to the entire abdominal pelvic cavity, whereas the male's is not. There was a question a moment ago when we were talking about the movement of the sperm. Does it move by pressure? And yeah, we will talk about the pressure changes and how it moves through there. So we will definitely talk about that as we work our way through that. So yeah, absolutely. We'll build to that, but we are going to absolutely do that. Excellent. So in males, we have this closed duct system, a set pathway that those sperm have to be made until it is released from the male reproductive system. And then, oops, no, I don't want my stars anymore. That ductwork system includes, but is not limited to, these four major structures, although as we'll see, there are some additional structures inside the testis as well. And so that ductwork includes the epididymis, the ductus or vas deferens, the ejaculatory duct, and the urethra. And remind me again, how many regions does the urethra have?
3.
3, excellent. There's the part of the urethra that passes through the prostate, which was called the prostatic urethra, excellent. There was the part of the urethra that passes through the levator ani muscle. What was that?
Membranous urethra.
Membranous, and the part that passes through the penis,
Spongy
which is the
urethra.
spongy or penile urethra, or cavernous too. Excellent. Perfect. And then, of course, there are going to be some accessory organs, primary glands, the paired seminal vesicles, the paired bulbourethral glands, and the singular the prostate gland. So there are these accessory glands in the system as well. All right, that is the basic anatomy, and we will talk about that. And then, of course, the external genitalia, the penis that we talked about earlier, and then the scrotum. Here we see it from this kind of, I don't know, longitudinal or frontal view. We see here we have the nice sagittal view that shows the relationship of these structures as well. And again, we got the advantage of seeing this when we were talking about the bladder as well, because obviously the bladder, many of these structures are part of the urinary system as well as the reproductive system. All right, let's start first with the housing for our testes, and that is the scrotum. The scrotum is a sac of skin and superficial fascia that is located outside the abdominal pelvic cavity and basically beneath the root of the penis. There are many biologists who believe that our primary function as an organism is to produce offspring, to propagate the species. If that is our one true vital function, then why in the world would you put the structures that are responsible for that outside the protection of the human body, right? Other than the fact that it can get you $10,000 on America's Funniest Home Videos, what's the point in having the testes located outside the body in the scrotum? Absolutely, it is to regulate temperature. As it turns out, the testes are quite capable of producing many, many millions of sperm. However, for those sperm to be viable when they are produced, they have to be kept at a temperature outside or lower, about 3 degrees lower than our core body temperature. So the whole purpose of having these paired testicles located in the scrotum outside the abdominal pelvic cavity, outside the human body, is to help to maintain temperature. And we'll see there's some specialized structures that are going to assist in that. The scrotum, again, is a pouch-like structure that is homologous. Let me write that word down. What does homologous mean?
Like one big structure?
Not a bad guess. It is similar to, homo does definitely mean one. So you're kind of on the right idea. Any idea what homologous means? Anyone taking genetics yet? Might have talked about this a little bit in micro. All right, so the same, but the same what? So the scrotum is what we call a homologous structure. From a biological standpoint, we all start as a, egg and a sperm that come together. And when that ova is fertilized by the spermatozoon, what do we call that structure that is formed as a result of that? Anybody know? What do you call a fertilized egg?
Oh, no, no.
I'm sorry?
Zygote?
Yeah, it's a zygote. Absolutely, that's zygote. All right, that zygote is a single cell that divides into many cells and eventually becomes a person. And it turns out the default setting for person is female. So if you just leave these things alone, then what you get as a result is a female. However, The males have that opportunity to provide a Y chromosome, which has some special epigenes on it. And those epigenes cause transformations to occur to deviate that growing ball of cells from the default setting of female to the alternate setting of male. What that means is that there are many bundles of cells, and those bundles of cells in women become one thing, and in males, they become something else. So basically, those two structures come from the same starting cells. When that occurs, we call them homologous structures. The scrotum is the homologous structure that in females become the labia majora. The outer folds of skin that form the boundaries of what someone mentioned earlier, the uvula or the vulva. That space covered with hair, thick keratinized skin. And so what basically happens is those two, the cells, the bundles of cells that become the labia majora, basically extend down and come together to form the scrotum. And one of the ways we see that is when these cells come together and form from the two sides, there is a raised ridge. I don't know how I'm going to do this now. There is a raised ridge that is found along the midline, basically forming a midline septum structure known as the raphe. Normally, when we're sitting in the classroom and we're not dealing with COVID, what I encourage you to do is to grab your nearest male, and if it happens to be yourself, then grab yourself and examine your scrotum, and you will see that there's this raised ridge along the midline. Normally, I tell you, if you don't have a conveniently located male, I encourage you to go to the nearest bar, walk up to a gentleman at the bar, and ask if you could see his raphe. And most of them are going to be more than willing to oblige. Of course, now we're not allowed to go to bars, so I don't know how you're going to do it. Do a web search for raphe. Maybe that'll do it for you. All right, just don't get the children's singer. That's not the raphe we're talking about. All right, but there's this raised ridge along the midline of the scrotum where basically those two labia, that homologous structure, comes and fuses and forms together. All right. Now, as we mentioned, this structure is involved in maintaining the testes outside of the abdominal pelvic cavity, because to produce viable sperm, and again, by viable sperm, I mean ones that are capable of fertilizing an egg. So when I say viable, I mean, again, you can have cells that are going to be sperm shaped if it was inside of your body, but they would not be capable of fertilizing that egg. They would not be capable of producing offspring. Now, there are several structures that help. Oh, great point. Typically, the testes descend into the scrotal sac prior to birth. Usually, we're starting around age 7 months of development. However, it isn't entirely uncommon for one or both testes not to descend. Originally, doctors weren't concerned about this at all, because after all, they're not going to need to use those until they're married and have college degrees, right? That's when you start producing offspring. At least that's what I told my kids. They have to be married and have college degrees before they can start producing offspring. So they're not going to be using those testes for a while. However, what they found is that a failure of the testis to descend, if that testis stayed inside the abdominal pelvic cavity for a prolonged period of time, what would happen is that it dramatically increased their likelihood of testicular cancer. So now, after about the first month, if the testes still have not descended, then the doctor will go in and surgically descend it into the scrotal sac, just to limit that potential for the increased risk of testicular cancer. All right. All sperm, and again, when you think of it this way, you've got an assembly line that is making somewhere on the order of 500 million sperm during the course of the day. Is every single one of those going to be a winner? Right? I mean, you'd have, as good as our quality control is, yes, exactly. Most, but not all of them will be viable. And there are many factors that will influence that as well. And we'll talk about some of those when we get along those lines, but there are going to be things that can influence and increase the likelihood of having more viable or less viable sperm. All righty, when we look inside the testis, as we can see here, there is an extension of the peritoneum. After all, this started in the abdominal pelvic cavity and then descends out from there. And so what happens is, not surprisingly, we have a double layer. Let's do that, and let's do that. A double layer of serous membrane that lines this space. Notice we have a parietal tunica vaginalis that wraps around the outer surface. And then we have a, so there's that around there. And then lining the surface of the testis, we have a visceral tunica vaginalis. So we have a serous membrane, again, producing serous fluid, that little bit of transudate to lubricate that space inside there, allow a little bit of freedom of movement. That tunica vaginalis is wrapped around and supported by a large fibrous structure known as the spermatocord. As you can see from this illustration, the spermatocord passes through an opening in this large fibrous white structure made up of parallel collagen fibers that goes from the superior anterior iliac spine to the pubic tubercle. Any idea what this structure might be?
Longitudinal ligament.
It is indeed a ligament, but what kind of ligament?
Inguinal, oh yeah.
There you go, the inguinal ligament, absolutely. And notice there is an opening in the inguinal ligament known as the inguinal canal. Now, in females actually have an inguinal canal as well, but in females, it is a potential space. Can anybody remind me what I mean when I say potential space?
Not space exactly.
Well, true, but what does that mean? Can you be more precise?
It's filled with some kind of fluid.
It isn't so much that it's filled, although you have the right idea. It's not a space. So the example I like to give, because I think this makes sense. If I take my hands and mash my hands together, when I'm mashing my hands together, do the two hands become one structure?
No.
No, of course not. However, is there a space in between them?
Yes.
Is there really? What could you put in there?
Yeah.
Could you put anything useful in that space between my hands right now?
No.
Maybe you could get a dime in there, but if I pushed hard enough, I could probably stop you from even sticking a dime in there, right? So there is the potential for a space there, but there's not a space. And basically that's how it is in female. Basically in females, it's a closed potential space, but in males, it's an opening that the spermatocord passes through. The spermatocord surrounds and protects all of the structures going into and out of the testis. There's got to be blood vessels going into and out of it, nerves going into and out of it, right? There's got to be the tube, the ductus deferens that carries the sperm out of it. And so it surrounds and protects all those things. Of course, that does mean that in males, this is a weakness. This is an opening to the abdominal pelvic cavity. And if the pressure in the abdominal pelvic cavity gets too big, fluid, omentum, or even organs can be pushed out through this space, bulging out in that space. And what do we call that condition?
Is that hernia?
That's exactly what that is. That is an inguinal hernia. Well over 90% of all inguinal hernias are by males. Females very, very rarely get an inguinal hernia. But there is another weak space that both males and females are equally likely to get a hernia. And what is that other weakness with that weak space? What's the other type of hernia? Come on, guys. I know it's holiday week, but let's
Umbilical.
pretend we care. Absolutely. Excellent. Umbilical, the belly button, right? Unless you're Adam or Eve, you have a belly button. And as a result of that, that is a weakness in your abdominal wall, where, again, too much pressure inside the abdominal pelvic cavity can force fluid, omentum, or even like a piece of the small or large intestine out that weak spot that we call an umbilical hernia. And since both boys and girls have belly buttons, they are equally likely to get umbilical hernias, whereas inguinal hernias are almost exclusively males. All right. Excellent. So we did that. Now, as I mentioned, the goal of keeping the testes outside the body is to maintain the appropriate temperature.
Excuse me.
Yes.
May I ask you about the hernia?
Yeah, absolutely.
My son, when he was like 2 months and a half, he had inguinal hernia. What do you think? What could cause that?
So in infants or young children, it's not that uncommon. Again, you got to remember the muscular wall isn't necessarily as strong. The ligaments aren't necessarily as strong. They bend themselves into, they can be very rough. They can be very tumble. And again, with small bodies, small changes in volume can cause more dramatic changes in pressure. So it's actually not uncommon at all for young males to get an inguinal hernia early on. And it's typically something they fix surgically, and it usually isn't something that causes any kind of long-lasting concern. So again, just morphologically, it tends to be more common in some people than others. But it isn't necessarily that there was anything wrong. It's just one of those things that happens.
But he cried a lot. They told me that it could be because of that, because he cried, so pressure was like increased inside.
Yeah, exactly. Anything that increases the abdominal pelvic pressure in the abdominal pelvic cavity can cause a hernia. In fact, and again, it's one of those statistics that is hard to get true numbers on, because not a lot of people are willing to admit it. But a surprising number of both inguinal and umbilical hernias occurred during defecation. If you think about it, defecation is one of those times where you're dramatically increasing the pressure in your abdominal pelvic cavity. And so as you're increasing that pressure in the abdominal pelvic cavity, that can occur. So some studies have shown, have speculated that as many as 1/3 of all hernias occur during defecation. But like I said, a lot of people aren't willing to admit it, so that they were, sitting on the toilet when they got their hernia. So it was, no, I was doing something manly, like moving my desk around or something like that. So yeah, the actual number is not fully known. But yeah, anything that increases crying would be a great example. Absolutely, those kind of things. Anything that increases the pressure in the abdominal pelvic cavity can increase the likelihood of something like that happening.
Thank you.
Yep. All right. So again, to produce viable sperm, we need to maintain temperature. And so if you notice, and actually let's stay here from the picture. Well, let's stick with the picture, and then we'll go for the pretty words. There are 3 structures that help us to maintain the temperature in the testes. Because while it has to be 3 degrees cooler than core body temperature, the problem is the outside world's temperature changes dramatically. And so we need to be able to dynamically adjust the environment to keep a stable temperature. And there are 3 main structures in the scrotum that allow that to occur. The first, as we see here from the illustration, is a layer of smooth muscle. And this layer of smooth muscle lines the outer wall of the scrotal sac. This layer is what is known as the Dardis muscle. The Dardis muscle, as I mentioned, is smooth muscle. So what does that tell you?
Involuntary.
All right, gives us involuntary control of that, something that we don't have to, we can't consciously control. It's controlled by our autonomic nervous system and by hormones. And basically what our Dardis muscle does is it can change the shape of the overall scrotal sac. If you think about it, much like the diaphragm, it is a curved muscle. And as we know, when a curved muscle contracts, what happens to the curve of that muscle? It flattens out, exactly. So basically, if the temperature is too cold, then it can contract the muscle, shortening the scrotum and bringing the testes up towards the abdominal pelvic cavity, bringing it closer for warmth. If, on the other hand, temperature increases, the muscle can relax, becoming more and more curved, allowing the testes to descend further away from the core of the body. So our smooth muscle, Dardis muscle, allows us to change the shape of the scrotum to be able to help to regulate temperature. But notice also there is a second muscle here, so let's change colors for that. We'll use blue. This muscle wraps around the testis and anchors up to the pubis inside of the spermatocord, as you can see here. And this is known as the cremaster muscle. The cremaster muscle happens to be skeletal muscle. What does that tell you?
Voluntary.
It can be voluntarily controlled. Now, does that mean that every single boy in this class is constantly monitoring the temperature of their testicles to decide how much to descend or ascend their testis inside of the scrotal sac? No, of course not. Not any more than all of you are voluntarily thinking about when you need to blink. It's been 3 seconds. I should blink now. And I should probably blink again. Oh, and I have to contract my skeletal muscle so I can breathe in and out. Open, then another blink, right? So obviously we have reflexes that control these things, but the same way that reflexes control my blinking and I can blink, reflexes control my breathing, but I can voluntarily control my breathing. Reflexes control the cremaster muscle, but also while you are asking that male that you've captured at the bar to show you his Raffi, also ask him to flex his cremaster muscle for you. And he should be able to voluntarily elevate and descend the testis in the scrotal sac. And absolutely, exactly, the scrotum changes with the weather, changes with the seasons, absolutely. All right, so we have this voluntary skeletal muscle, which can be voluntarily controlled, that helps to ascend and descend the testis within the scrotal sac. All right. Awesome. All of that is great. We have it outside the world in this unprotected environment. We have muscles to move them around all over the place. But we still have this major issue. And that major issue is that the testes make spermatozoa. They make the sperm. And they make a lot of sperm. Like I said, 500 million a day or something crazy like that. So not surprisingly, that requires a tremendously high metabolism. Quite frankly, we're using so much energy to produce sperm. Exactly. I'm surprised we're asked to do anything else. We should just be able to sit on our chairs and make sperm, and that should be enough for everybody. But to have all of that metabolism obviously need lots of energy. And of course, that means lots of oxygen and nutrients. And of course, how are we going to get those oxygen and nutrients to the testes?
Blood supply.
Blood supply. Remember back in the cardiovascular system, we talked about how let's actually do it this way. We talked about how that testicular artery that gonadal artery, the testicular artery, was larger and longer than the ovarian artery because the testes are much more metabolically active. However, the other thing we talked about in the cardiovascular system is that blood is 2 to 3 degrees warmer than our core body temperature. And if our core body temperature is too hot for the testes, then what's going to happen with this massively hot blood that is going to the testes? It doesn't matter how far away they are from the body. If we have 100 degrees Fahrenheit blood pouring into them, Right. You have a major issue of it being too blood. Ridiculous. That's a great question. Let's come back to that in a second. Let's deal with this first. So we have this hot blood coming, and that could be a major issue. Luckily, we've already learned how to fix this. Remember, like in our urinary system, we talked about those heat sinks. Right? We talked about using that counter current mechanism to be able to draw the heat off. And the way we do that, as you can see from the illustration, is there is this elaborate network of veins wrapped around the testicular artery. This big elaborate structure is what is known as the Pampiniform Plexus. And this Pampiniform Plexus is basically an elaboration of the testicular artery. What happens is we have cool blood entering into the Pampiniform Plexus and traveling back across the testicular artery. And as it does that, it absorbs the heat. And so basically it warms up as it works its way back. Of course, it's stealing that heat from the arterial blood on the way down, so that by the time that arterial blood reaches the testis, it is now cool blood. And that cool blood now can not damage, not affect the function of the testis. And then that cool blood on its way back draws up the heat and helps to be able to allow it to function, warming the blood back up as it goes back to the heart. So that countercurrent mechanism, that heat sink that we talked about in the urinary system, we're taking advantage of it here as well, with that big draping blood vessel. Now, Radhika, I've not heard of a varicose vein in the testis before. Again, reproductive system is not my area of expertise. Typically, varicose veins occur in areas where there is dramatic increase in pressure. So I guess if maybe someone receives some type of injury or something along those lines, that could cause an increase in pressure, or could cause an edema in that area, which could cause a varicose vein to occur. Basically, a varicose vein is when the vein becomes dilated because of a failure of the valves to be able to function properly. And so you get an accumulation of blood in that area. It does cause that area to be congested. Usually when they happen in places like your legs, it is superficial, so there are plenty of other routes that the blood can take back, so it doesn't impair function so much. However, here in the testis, it's much more limited. There's only this one major blood vessel that comes out. So if it became varicose for some reason, and again, I can't off the top of my head know why that would happen or how that would occur, but theoretically, if it did occur, then yes, that could be a concern, because you could get increased edema in the testis that could cause swelling, it could cause congestion, a decrease in function. So I could see where things like that could potentially be a problem. Again, it doesn't necessarily have to be, and I guess it would have to be how elaborate and again, also where it was. Remember, there's also going to be superficial blood vessels in the scrotum. If it is a superficial blood vessel in the scrotum that has become varicose and is obvious from the outside, then again, it's going to be more superficial, it's going to be more appearance, but it shouldn't affect the function of the testes. So it also depends on where it was. All righty, good questions, excellent. All right, so I've done it here with the picture, but let's do it with the pretty words. Again, we have that Dardis muscle, wrinkles the scrotal, changes the shape of the scrotal sac. That Cremaster muscle, which elevates and descends, and then those Pampiniform Plexus. And I think I have another picture here which shows them nicely as well. So again, here we really get a sense of that elaborate network of that vein over the top of the testicular artery coming in. and then the nerves and other stuff that are there as well. Again, here we see the cremaster muscle inside of our spermatocord, wrapping around the layers of our tunica vaginalis, both parietal and visceral, that Dardis muscle line in the scrotum, again, and that midline septum, which again, as we mentioned, not only there's an internal midline septum, but remember the one thing they're really not showing us is that raised ridge that would be the raphe on the surface of this. All right, but yeah, there is that septum separating the two testes from each other. Each of them have their own individual compartments. All righty, questions on that? All right, then let's talk a little bit more about the testis. Once we get the anatomy down, we can start talking about the function of it. As we look close at the testis, as we can see, as we mentioned, there is that outermost layer that is the visceral tunica vaginalis. Tunica vaginalis. But the bulk of the testis is made of this fibrous white connective tissue structure known as the tunica albuginea. Now the tunica albuginea, and again, I don't have anything that I can draw white on, so let's cheat and use, I'll just use blue because that's the color that I have. Not only does it form the outer structure of the testis, the bulk of the testis, but as you can see, there are deep invaginations of the tunica albuginea. These deep invaginations are what we call septa, the singular being a septum, and it divides the testis into individual lobules. So here I have a small single individual lobule. And each testis is divided up into somewhere between 250 and 300 lobules. Now, each of these lobules inside of them, as you can see, contain a coiled tubular structure that we are familiar with from the endocrine system, that is the seminiferous tubules. Each of these seminiferous tubules are, 1 to 4 seminiferous tubules in each one of these, and each one of these seminiferous tubules are about 3 feet long. So if you think about it, if you had 4 seminiferous tubules in every single one of your 250 lobules, and each one of them was 3 feet long, if you would take them out and line them out end to end, it would hurt a lot. All right. Now, someone mentioned earlier the movement of the spermatozoa. As we know from 430, spermatozoa are the only cells in the human body that are capable of self-propulsion. They have a special organelle, and what is that special organelle again?
Flagellum?
Yeah, the flagellum, absolutely. And that flagellum basically gets moved in a whip-like motion to propel itself through space. We haven't talked about the anatomy of the sperm yet, but what I'll tell you is there's a motor in the midpoint that rotates that flagellum around. And basically it ratchets around like a gear. And every single ratchet of that gear requires 1 ATP. So to spin that tail around to propel itself through space requires a tremendous amount of energy. So when it comes to moving the sperm through the ductwork system of the males, do we let the sperm move themselves?
No.
No, it would be an incredible waste of energy. Right, incredible waste of ATP, incredible waste of resources and things like that. So we are going to use pressure, fluid pressures, as someone mentioned earlier. The other thing we are going to use is smooth muscle contractions. In some of the larger, oops, hit my computer. In some of the larger structures, they will actually use peristalsis. But inside of our seminiferous tubules, or more specifically, I should say, outside of our seminiferous tubules, are some very thin, smooth muscle cells called myoid cells. And these myoid cells basically squeeze the seminiferous tubules. The seminiferous tubules make fluid as well as sperm. And so when they squeeze, that decreases the volume, increases the pressure, produces a pressure gradient on that fluid, and that fluid is forced out of the seminiferous tubule and carries the sperm with it. It is like segmentation in that it is a contraction of a circular muscle, but segmentation, remember, is not about propulsion. It's about moving the stuff back and forth. This is really a one-way movement. So it's basically squeezing the tubes to get it, like squeezing a tube of toothpaste. You're supposed to. You start at the base and you squeeze your way up to get it out. And that's kind of what's happening here. Great question. In fact, self-propulsion of the sperm doesn't get it out of the body. Typically occurs once it is out of the body. Typically, it's not going to start swimming until it is expressed out of the male ductwork system into the female vaginal canal. They're responsible, with actually some help of the female reproductive tract, to get to the egg on their own. So yeah, they don't really start swimming until the ejaculatory process. Excellent. Here is some anatomy we have seen before. A cross section through our seminiferous tubules. Notice some of the structures we see here. Here we see some blood vessels. Here we see our seminiferous tubule containing all of the maturing cells that are becoming the sperm. And someone remind me again, what are these cells out here called? No, not the myoid cell, not the nurse cells. Interstitial cells, excellent. And what again are the function of those interstitial cells? They produce the androgens, absolutely. Remember, it's the interstitial cells between the seminiferous tubules that are responsible for producing our hormones, those androgens. Excellent, excellent. So again, each is about 3 feet long of these seminiferous tubules, 1 to 4 of them per lobule, 250 to 300 lobules. Again, a half mile of seminiferous tubules per testis, and their job is to make our spermatozoa. Develop cell-shaped cells, stem cells, into sperm-shaped cells. All right, that's their job, to go from stem cells to sperm-shaped cells. And as we talked about, those interstitial cells are responsible for producing our androgens. Now, here's a nice illustration that does a good job of showing this, and this allows us to see some of the other things that we have talked about. For starters here, we can now see those myoid cells, that thin single layer of smooth muscle cells on the outer surface of the seminiferous tubule. Here we see some interstitial cells that are in the septum, the connective tissue septum between the lumen. So this would this septum is of course part of the tunica albuginea. And in that tunica albuginea septum, we have the interstitial cells and blood vessels and things along those lines. The cells in blue and black represent the different stages of development for our sperm. going from stem cells starting at the base. Wow, that is way too big. Starting with stem cells, starting at the basal surface where the myeloid cells are, and maturing into sperm-shaped cells by the time they find the lumen. But notice there's this shaded blue, this shaded purple region with these triangle or diamond-shaped nuclei. These purple shaded regions represent those nurse cells, or the Sertoli cells, or remember they're also called subcentacular cells, the support cells that play a huge role in the maturation and development of our spermatozoa. So we talked about the interstitial cells of Leydig that produce the androgens. And again, we see those outside of the seminiferous tubule. Notice this one shows the myeloid cell much more nicely. And here, very, very clearly, we can see some examples of those nice, prominent, triangular or diamond-shaped nuclei that are the nurse cells. These nurse, oh, there we go. Love these pictures from your textbook. Great electron microscopy view. See the blood vessel. Here we see that seminiferous tubule, and here we see all the flagella, all the sperm-shaped cells located at the center of that seminiferous tubule. Excellent. Now, as I mentioned, if you're going to be producing 500 million cells every single day, you're going to need stem cells to do that. These are a unipotent stem cell. Remind me again what that means?
And make more than one kind of cell.
More than one?
No, just one.
Just one.
Oh, unipotent.
Stem cells, unipotent. These are sorry, that's a little small. There we go. Unipotent stem cells. These are stem cells that produce one and one thing only. The Spermatogonia are pretty easy to find because they're always going to be located along the outside edge of the seminiferous tubule. So here's a good example of one, and here's a good example of one. Here are some more. So there's one. So all these cells right along the outermost boundary, there's a nurse cell, are the Spermatogonia. Their job is to constantly divide, producing new cells. And like I said, the average male produces about 500 million sperm per day. Starting when? When do males start producing sperm? Puberty? Puberty, there you go. So what, around 13? When do they stop making sperm?
Never.
Well, okay. Well, then not never. I wouldn't say never. How about, but you got the right idea. Basically, there you go. I like that. 15 minutes after they die. Someone said 15 minutes after they die. Absolutely. Yeah. So you get guys like Hugh Hefner or guys like, all sorts of these other really old, rich guys, actors and stuff like that, having kids that they're too old and frail to be able to pick up. Tony Randall was a great example of that. One of those kind of guys. Absolutely. So 500 million a day, every day, 365 days a year for 50, 60, 70 years, something like that. I mean, the numbers are astronomical when you start thinking about them. And like I said, we have these nurse cells, Sertoli cells, subcentacular cells. All of these are appropriate names for this. And I like that previous picture, but I think this one kind of does a little bit of a nicer job of showing exactly what it is that these cells are like. As you can see, here's the nucleus of this cell that has a large amount of cytoplasm to it, wrapping around and supporting the cells. But notice it is forming tight junctions with the nurse cells next to it. Tight junctions, again, are going to limit the things that can get into and out of the seminiferous tubule. Great question. There can be it is not uncommon for there to be a slight decline in the amount of sperm and the viability of the sperm that are produced as a male ages. However, we've all seen on the news and other places plenty of examples of people even in their 80s and 90s producing offspring because, again, while it can decrease, they're still going to produce a significant amount, and there's probably a genetic predisposition to that. But yeah, there's plenty of, frail elderly men producing offspring. All right. Now, as I mentioned, these tight junctions form, and basically they form a barrier between what's going on outside the testis, and outside the seminiferous tubule, and what's going on inside. Because remember, these sperm are, as you've learned from the labster, hopefully, very, very special types of cells, unique cells. And the problem with unique cells is that your body doesn't necessarily like things that are unique. Do we necessarily want our antibodies, our immune cells coming in here and checking out these sperm to see if everything is okay? No, that would probably be a bad thing, absolutely. So these Sertoli cells play a huge important role in helping to form what we call the blood-testis barrier, protecting these unique immature cells that are being produced and stopping us from getting an immune response that attacks our testes. That in and of itself would be vitally important, but our Sertoli cells are actually the major cells involved in supporting sperm development. What was the hormone again that we said that regulates sperm development?
Testosterone.
What was it?
Testosterone.
No, what was the hormone? The hormone that is responsible for sperm production. If only we had learned the hormones in this class.
Luteinizing hormone.
There it is. Nope, someone's got it. Follicle stimulating hormone. Follicle stimulating hormone, remember, was the hormone we said that regulates control of sperm production. What follicle stimulating hormone actually does is it actually stimulates the nurse cells. And then the nurse cells provide nutrients and chemical signals to the sperm cell to stimulate the division from stem cell into sperm. Notice that transformation from cell-shaped cell to sperm-shaped cell causes a dramatic change in the cell. Basically, all sperm are genetic missiles, basically containing DNA and not much else. So this cell has to shed a massive amount of its cytoplasm. And does that cytoplasm go to waste? No. As it turns out, these Sertoli cells, oops. Oh, there we go. Okay, so it's at the end, so we'll come back to it. The Sertoli cells actually phagocyze that excess cytoplasm, storing the nutrients and using it to provide for the next cell that is dividing. So it helps to recycle those materials. Our nurse cells also produce the fluids. Remember, we talked about how it's fluid pressures by that myoid cell, which we can also see here. Notice this little pink cell right here on the outer layer is that myoid cell. So the fluids that are going to sustain the sperm and provide the fluid pressures to move it out of the seminiferous tubules. But our nurse cell also is responsible for producing hormones. In particular, our nurse cells produce the hormone inhibin. Remember, inhibin plays a vital role in inhibiting the production and release of follicle stimulating hormone. because as we said, even though we have to make 500 million sperm a day, the testes could do more, and more isn't necessarily better. So we want to regulate that process of sperm production, and inhibin plays an important role in that. Remember also, I keep saying that at the end of this process in the testis, we have sperm-shaped cells. Notice I don't say that there are spermatozoa in here, because truly spermatozoa are the mature cells that are capable of fertilizing the eggs. When these cells are produced, they are still immature. They actually have to leave the testis into that first tubular structure, the epididymis. And in that epididymis, they take 20 days to mature. And that maturation process requires androgens. Remember, way back in the endocrine system, we said androgens don't affect production of sperm. They affect the maturation of the sperm. So what that means is we have to get those androgens, and remember, I'll just draw it as a nice big A here. Here is that interstitial cell, and that interstitial cell is making the androgen, and we got to get that androgen to the epididymis. Well, what we can do is we have this fluid that is going to be carrying the sperm to the epididymis. So we can take this androgen and we could dump it into the fluid. What's the problem with dumping an androgen into fluid? Why don't you want to just take androgens and dump them into water? Well, but we want it to get in the blood. But do we really want androgens just straight into the blood, pour them like a bucket? Why not? You're absolutely correct, but why? What do we know about androgens? Hormones produced in the testes. What are they made of again? Cholesterol. They're lipid-based. And do we want to put lipid-based things just straight into water?
No.
No. So one of the other things that our nurse cells make is an antigen binding protein. This is a special protein that can bind the androgen. It actually binds several androgens and is water soluble so that these androgens can be transported to the epididymis to help in the maturation of the sperm. And then, as I already mentioned, these nurse cells help to digest and recycle the leftover cytoplasm that is shed in the process of making sperm-shaped cells. So like real nurses, nurse cells are true rock stars that wear many hats and serve many functions. So that should be easy to remember. Questions on that? It does not store a lot of nutrients, but when it's going to be functional, it is going to need a lot of nutrients. Absolutely. But when it comes to being a viable cell, right, does it need, mitochondria? Does it need a Golgi apparatus? Does it need a rough endoplasmic reticulum? Does it need a smooth endoplasmic reticulum and all those kind of things? No. Like I said, it pretty much just it's a biological missile, right? Shoot them off, waste them. When you got 500 million of them, right? None of them is going to be particularly important. You just throw out as many as you can and hope for the best. All right. Excellent. Excellent. All right. So Again, not my area of expertise. I would be surprised if there were fully functional third testes. I could see where you could have maybe some type of additional growth or something that occurred there, but I'm not sure if it necessarily would be fully functional and connected the same way the others were. But again, if anatomy and physiology has taught me nothing, then I'm guessing those, I mean, if you can have, they have mutations of, you know, fish with 3 eyes and things along those lines. So I'm guessing something like that could happen. My guess is that it's not a common mutation that occurs, but is it possible that something like that could have occurred? I don't see why not. Yeah. But as far as I know, it's probably not common. And like I said, I'd probably be surprised if it was fully functional. All righty. Time to talk birds and bees. As I mentioned, our goal now is to make these puzzle pieces and put these puzzle pieces together to make a person. This kind of takes us into the realm of genetics. And as we've talked about in this class many times, one of the things about anatomy and biology in general, but especially anatomy and physiology, and especially genetics, there is a large amount of vocabulary. So to be able to truly understand this process of mitosis and meiosis, which again, is the first time I've given that labster. I'm hoping having it ahead of time will help in this discussion because I find a lot of people are intimidated by this material and haven't looked at mitosis since 430. So I'm hoping that will help us to be able to tackle this in a timely fashion. But I do want to come at it fresh. So this is probably a good stopping point. Let's go ahead and take our first break now. Any questions before we take our first break? Go ahead. I'm a firm believer. The only bad question is the question not asked. Which question?
Okay, so when males get hit in their balls and we don't feel the pain in our scrotum, we feel it in our stomach or in our back. And I was wondering if you knew why we feel the pain in those areas instead of where we got hit.
Yes. So it's what we call referred pain. And there are many parts of the body that experience that. And the short answer to your question is, if you remember back in 430, one of the things we talked about in 430 is that most of our pain pathways are unmyelinated. Remember, while unmyelinated axons are slightly slower, they're much more consistent in getting that signal through. However, being unmyelinated, it makes it more likely that action potentials can be transferred to other axons. And so, like, for instance, one of the things when people have a heart attack, where do they often feel pain as a result of that heart attack? Anyone?
Back.
In their left arm.
Back or the left arm, right? They feel that pain in the left arm. That's one of the things you always see them in the movie, grabbing their left arm when they're having that heart attack because of that. That's another example of referred pain, right? When your stomach way down here hurts, you feel it up here, that heartburn, right? That's where it gets the name heartburn, because that referred pain that is coming up. So it's just another example of that referred pain. That large pain stimulus coming up there can be transmitted throughout that region, and it's a spreading of there. Although I would argue with you that while feeling it in your gut, in your abdominal region, if you get hit hard enough in the testes, you feel it in the testes. It's not an enjoyable feeling for the testes either. So, but yes, basically, like, because many pain pathways are unmyelinated, you do often see referred pains in regions like that. Yep, and that was a great question. Any others? All right, so we'll take our break then, and when we come back from our break, looks like it's 1:20, so we'll come back at 1:35, and at 1:35, we will tackle mitosis and meiosis. We'll start the recording at that part. All righty, see you guys in 15 minutes. All righty. Let's start. our goal next is going to be to talk about the process of sperm formation. That process of sperm formation is called spermatogenesis. This process of spermatogenesis involves three processes. The first of those processes is mitosis. Someone remind me again what happens in mitosis?
One cell becomes two cells.
Excellent.
Identical.
One cell becomes two cells.
Identical. Yeah.
Excellent. Two identical cells. And by that, I mean identical to the original and also identical to each other. So identical cells, and again, that is to each other and the original one. And how many divisions does it take to get one cell to become two cells?
You mean how many phases?
No, not phases. How many times does a cell divide in this process?
Once.
Yeah, so there's one division. And where one cell becomes two identical cells, identical to each other and identical to the original. Oops, this is different. I guess I needed to move that now. We'll sneak this down here for now. How is this different from meiosis? How many divisions in meiosis? Two divisions, excellent. And in those two divisions, one cell becomes what?
Four.
Excellent. Becomes four cells. Are these cells identical? No, four unique cells. Both from each other and from the original. Excellent. So first we have mitosis, then we have meiosis. But then after that, we still have to go from cell-shaped cells to sperm-shaped cells. And that process, and I guess I'll erase all of that now. That third process is called spermiogenesis. Spermiogenesis is the way that that cell-shaped cell is transformed into what we see here, a mature spermatozoon. That sperm-shaped cell. That is that genetic missile that we have talked about. All right, now when we discuss this process, we need some definitions. As I mentioned, when we talk biology, when we talk genetics, we need definitions. The first involves the number of chromosomes that a cell has inside of it. Right? Most of the cells of our body are what are known as diploid chromosomal cells that have a diploid number of cells. No, great question. So spermatogenesis is the process of forming sperm, yes. Spermiogenesis is one of the stages of spermatogenesis where we go from cell-shaped cells to sperm-shaped cells. So the cell changes its shape, changes its morphology, changes its function, because when you change your structure, you change your function. Okay, so it's the third step of spermatogenesis. Excellent. All right. So most of the cells of our body have a diploid number of chromosomes. What is the diploid number of chromosomes in a human? Excellent, 46 chromosomes. Or, oops, I did that all right. But again, diploid refers to what? You got the right idea, Alex. Diploid refers to two pairs, because what happens is what we really have is 23 pairs of unique chromosomes. One of the important things to remember is a chromosome is a collection of DNA and proteins. That DNA and protein folds up And actually, let's just, I won't even bother trying to draw the squiggly. We'll just do it this way. All right, it's just this big, huge collection of DNA and proteins. That loose chromatin is folded up into a tightly packed chromosome. And the important thing to remember about these chromosomes is each one of these 23 pairs contains unique information. So for instance, let's say, and again, I know none of this is correct information, but it doesn't matter, it's the point. Chromosome 1, for instance, contains the information in this region right here for handedness, whether you're left-handed or right-handed. All right. And then it has the information in the DNA right here, for instance, for blood type. And then it has the information, for instance, right here for a predisposition for heart disease. Right. So all of this information is on chromosome 1, and chromosome 2 will have completely different types of information. So chromosome 2 has different information on it. So for instance, it has the color of your eyes, or whether you can roll your tongue, right? Or how tall you're predisposed to be, those types of things like that. So each of our 23 chromosomes contain unique information. But as Alex pointed out, we have pairs of chromosomes. So this one right here, for instance, is chromosome 1 from dad. But we are also going to have a chromosome 1 from mom. And that chromosome 1 from mom contains similar information to what we got from dad. So it is going to have handedness in the same location. It is going to have blood type in the same location. It is going to have the predisposition for heart disease in the same location. However, while it has the same type of information, is the information necessarily going to be identical? No. So for instance, over here, dad's chromosome 1 could give us the gene that gives us the A antigens on the surface of our red blood cells. Mom's could give us the B antigen. And so if this were your 2 chromosome 1s, what would your blood type be? AB, right? If you got an R from dad and you got an L from mom, what would your handedness be? Nope, you'd actually be right-handed. Right-handed is dominant. So again, we'll get into genetics at the end of this, but the point is it can contain different types of information. These 2 chromosomes, these paired chromosomes, are what we call homologous chromosomes. So everybody has 23 pairs of homologous chromosomes. Okay, so far so good? Excellent. All right, so now here's the key to this. So we've got this part so far with all of this with the different chromosomes. Now this is just for humans. All right, ducks have a different number of chromosomes. Plants have a different number of chromosomes. Everybody has a different number of chromosomes, but they always get paired chromosomes because anything who reproduces sexually gets half of their chromosomes from mom and half of their chromosomes for dad. So the way we represent our diploid number of chromosomes is by saying 2n. 2 represents that you have 2 copies of it, one from mom and one from dad, and n is the number of unique chromosomes. So for humans, n equals 23. There are some plants where n could equal 96. right? They have totally, plants have crazy numbers of chromosomes in some of their cells, but n equals 23. So as we talked about, our diploid number is 46. So some fun algebra there. Does that make some semblance of sense? I'll pretend that it does since everybody's quiet. I want to leave this stuff, but I need to get rid of all of this. Excellent. So there you go, 2n. However, when me and my wife decided to have kids, did I take a cell, one of my cells with 46 chromosomes, and her take her cells with 46 chromosomes, smash them together, get a kid with 92 chromosomes, and now she can see through walls and has super strength and all those kind of things? No, of course not. When we want to produce offspring, we need to make sure those offspring have the same number of chromosomes as we do. And so our gametes have to be haploid. Hap, of course, refers to half. So in this case, it's just 23 chromosomes. But is it just some random collection of those chromosomes? So I throw 2 number 1s and 1 number 3, but not a number 2. No, you get 1 copy of each unique chromosome. And so, not surprisingly, we represent that by just n. So my n and my wife's n. Exactly. So I give 1 chromosome 1, my wife gives 1 chromosome 1. Right? I give 1 chromosome 2, my wife gives chromosome 2, and we put those together. So each of us make haploid cells that have 1 copy of each of the unique 23 chromosomes. And that is n. All right. And of course, the only haploid cells are what? The gametes, exactly. The eggs and the sperm. Excellent, excellent, excellent. All right, so these are our homologous chromosomes. We talked about those. All right. But we also have to go back to some of our vocabulary that we talked about in 430. If you remember in 430, when we talked about mitosis, we know that if, oh, and let's go one more step before we do this. All right. So as we learned in mitosis or in the cell cycle, if one cell is going to become 2 cells, and both of those cells are going to have 46 chromosomes, right? Because that's what mitosis is. Mitosis, we have 1 cell become 2 cells, and we want both cells to have 46 chromosomes. How do we do that? How can we divide 1 cell into 2 and have both cells have 46 chromosomes? How are we able to do that in mitosis? Okay, during the concept of cell replication and division, exactly. What do you mean by replication? Make a copy of the DNA, absolutely. We have to make a copy of the DNA. We have to replicate the DNA. And in fact, we replicate that DNA first. Does anybody remember when DNA replication actually takes place in the cell cycle? There you go, the S phase of interphase. When our DNA and proteins are still loose chromatin. So if you remember in mitosis, when that loose chromatin folds up to become a tightly packed chromosome, it doesn't just do it by itself. There are 2 identical copies of it, and those 2 identical copies are held together by a special protein. This whole thing in its entirety, bloop, 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 we call 1 chromosome. All right. So this is 1 chromosome. but also this thing by itself is one chromosome. I should go green, be consistent. So how do we tell that apart? If this is one chromosome and this is one chromosome, and we have 46 of these and we have 46 of these, how do we tell them apart? Well, we have another nomenclature, and your book's not as good at using this. So that's why I want to emphasize this now. When the DNA is replicated, we would indicate that by using the nomenclature of 2NR. That R indicates that it is replicated. So 2N is one copy of each of the chromosomes. 2NR is when you have two copies of each of the chromosomes. All right? So this would be, N, this would be NR. Now, we need some more vocabulary. When we have these two identical copies stuck together, what is the term we give for each of the individual copies?
Sister chromatids.
Sister chromatids, excellent. And what is the name of the special protein that holds them together? Centromere. Excellent, excellent. I like this. Getting far more answers than I normally get to this. I like that we did the Labster first. I may do that even when we're back in the classroom. Excellent, excellent, excellent. So we have these. One chromosome, that one replicated chromosome is made up of two sister chromatids connected by a centromere. And again, we would indicate that as 2NR. So actually, let's do that. I want to change that. Because I want to really beat that horse dead. Excellent. So one replicated chromosome is comprised of two sister chromatids held together by a centromere. And that would be 2NR. Our homologous chromosomes, remember, are the two copies. One from mom and one from dad that contain the same type of information. So like I said, chromosome 1 from mom and chromosome 1 from dad would be our homologous chromosomes. All right. Chromosome 1 and chromosome 2 are not homologous because they don't contain the same type of information. But chromosome 1 from mom, chromosome 1 from dad, those are homologous because they contain the same types of information. Excellent, excellent, excellent. All right, so that is our vocabulary. Any questions on this? This is gonna, if we understand this vocabulary, it's gonna help us going forward. If we don't, it's gonna get a lot worse. All right, stunned silence means I understand it and make the test harder. Perfect, excellent, let's work on. So let's talk first about mitosis. Again, this should be pretty easy because you guys just had to do this. So remind me again, and I'll do this simple. We won't even bother drawing it out. Mitosis. How many stages?
Five.
Yeah. How many?
4, but there are early prophase and late prophase.
So 4 stages, but sometimes we take that early prophase and late prophase and divide it into 5, where that we have prophase and prometaphase, but we'll stick with 4 for now. And again, if we stick with 4, what's the mnemonic to help us to remember that?
Prophase, metaphase, and
PMAT.
There you go. Prophase, metaphase, anaphase, and what was the last one?
telophase.
Telophase, there you go, exactly. Just remember what you have in the bathroom. You have a PMAT in the bathroom, and that'll help you to remember those 4 stages of that. Remember also, of course, that is just the division of the nucleus. So again, we also have to have the cytokinesis to divide the cell in half. Excellent. Real quickly, remind me what happens in prophase. Excellent. So be careful. Yeah. So our chromatin folds up into chromosomes. Excellent. What else? A nuclear membrane, nuclear envelope breaks down. Excellent. What else? Excellent. And we have the two centrioles that migrate to the opposite sides of the cell, to the poles. And remember, they start producing those spindle fibers. And those, what do those spindle fibers do? They stretch the cell out. But what's the other thing that those spindle fibers do? Well, they're not going to pull them apart yet, but you got the right idea. They're going to connect to the chromosomes. Excellent. Connect to the kinetochores. Excellent. So a whole lot going on in prophase, which, as we mentioned, is why they've started talking about early prophase and late prophase, and even taking prophase and dividing it up into prophase and prometaphase, because there's a lot going on there. Right? What happens in metaphase? Right. Individual chromosomes line, and again, these are replicated chromosomes, so we should probably mention that. Line up on the metaphase plate, which is the equator. Which is the equator. Excellent. And again, they do it independently. Each one for themselves. All right. What happens in anaphase?
Chromatids are separated from each other and pulled to opposite poles.
Excellent. The centromeres inactivate. And when they inactivate, our sister chromatids separate into what? What do our sister chromatids become? There you go, daughter chromosomes. Excellent, into daughter chromosomes. And then, as you just mentioned, those daughter chromosomes are pulled to the opposite sides of the cells. And that leads us to telophase and also cytokinesis. What happens in telophase? Let's do telophase first.
The identical sets of chromosomes at the opposite poles of the cell.
Yep.
They uncoil and resume their chromatin form.
So excellent. We uncoil our identical chromosomes into loose chromatin again. All right, excellent. What else?
New nuclear envelope forms around each.
Two.
Yeah.
Right, because one for each. New nuclear envelopes forms. All right, excellent, right? Around them. And then with cytokinesis, of course, then the cytoplasm is divided. And we have two new cells. Yep, exactly. And I like what Alexa pointed out. Yeah, in many ways, telophase is kind of prophase in reverse. Our loose chromatin is condensed down into chromosomes, whereas here our chromosomes are uncoiling into loose chromatin. Our nuclear envelope breaks down in prophase. Our nuclear envelopes, plural form, in telophase. Excellent. And again, one division gives us two identical cells. Now, the good news is meiosis is very similar to mitosis, where it has these same four stages: prophase, metaphase, anaphase, and telophase. But remember, in meiosis, division has to occur twice. So the way we identify those is by going through those processes twice. The first time, it's prophase 1, metaphase 1, anaphase 1, telophase 1. And the next time through, it's prophase 2, metaphase 2, anaphase 2, and telophase 2. If we are comfortable with mitosis, then I want to spend the time going over meiosis. And again, hopefully this should be familiar from you from 430 and through the Labster. So any questions on mitosis before we compare it to meiosis? And again, the one last thing I want to point out is here we have one division for two identical cells. And again, identical to each other and identical to the original. All right, nobody's spoken up, so let's do meiosis. Again, for meiosis, our goal is to have two divisions. And from those two divisions, we get 4 unique cells. Right? Unique from each other. Well, unique from the original and from each other. All right. Now, again, there are 2 divisions that take place, and the first division is Meiosis One. And as we mentioned, Meiosis One is going to have 4 stages in it: prophase 1, metaphase 1, anaphase 1, and telophase 1. Meiosis One is the part of the process that is most different from what we see in mitosis. Because if you think about mitosis, is about dividing the replicated chromosomes. But that's not what meiosis is about. Meiosis is really the division of the homologous chromosomes. We start this process because, again, when we start this process, we've already gone through S phase. So we start with one cell that is 2NR, diploid and replicated. And at the end of the process, we have 2 cells, and these 2 cells are both NR, haploid and replicated. So we go from diploid and replicated to haploid and replicated. Let's talk about how this would occur. We'll do it simple by starting with a really basic cell. If I were to try to draw a human cell here and try to cram 46 chromosomes into it, that would be nigh impossible, all right? But what I can do is start with a very basic cell. And start with a basic cell that says, let's say, has 4 chromosomes in it. And we'll do them by color. Chromosome 1 is dark red from mom and light red from dad. Chromosome 2 is dark green from mom and light green from dad. Actually, let's go ahead and do 3. And chromosome 3, dark from mom and light from dad. Now, all of these 3 sets of homologous chromosomes, right? So again, our 2N number would be, my N number here would be 3. So my total number of chromosomes would be 6. So this cell has 6 chromosomes. I want to divide this cell twice. And at the end of dividing it twice, I want to have 4 cells that are haploid in number, only have 3 chromosomes. Now, to do this requires 2 divisions. Meiosis One has prophase. One. And again, like most things, they use Roman numerals for these types of things. Metaphase 1. Anaphase 1. And telophase. Now there's still prophase, metaphase, anaphase, and telophase. So there are still going to be some similarities in these processes. Let's go ahead and draw our cells. In prophase 1, just like in prophase of mitosis, one of the first things that is going to happen, actually, you know what? Let's do it this way. Rather than trying to fit it all on the page, I will split it up. And that'll give me room to write, because I'm clever. All right, excellent. In prophase 1, we are going to see the things that we expect to see. The breakdown of the nuclear envelope. Now I want this to be smaller. Breakdown in the nuclear envelope. Our folding of our chromatin into chromosomes. And we expect to see the moving of the centrosomes to the poles. So we are still going to have our centrosomes at the poles. And we're still going to have our chromosomes. And again, remember, these chromosomes are going to be H-shaped, although I'll draw them as X's because it's more convenient. Oh, there's an X shape. And here's an X shape. And here's an X shape that are going to form. So all of those kind of things. And of course, the breakdown of the nuclear envelope. I guess I can give little nuclear envelope pieces, although that'll just confuse things. Let's form our spindles. But here is where one additional thing is going to happen. Here in prophase 1, we are going to form structures that we call tetrads. The big new event that is occurring here is that we are forming tetrads. And what this basically means is that our homologous chromosomes find each other. And they use a special protein called synapsin to basically zip themselves together. So rather than each individual chromosome being on its own, what is going to happen, oops, no, hold on, that can be black. What is going to happen is that chromosome 1 from mom is going to find chromosome 1 from dad. Chromosome 2 from mom or from dad finds chromosome 2 from mom. Chromosome 3 from dad finds chromosome 3 from mom. And our homologous chromosomes come together. So both homologous chromosomes with a special protein called synapsin zip themselves together. And when they zip themselves together, they form a tetrad. And so then the last thing we have to do is using our spindles, we attach, that's not how you spell spindles. Using the spindles, we attach our spindles to the tetrads. So let's actually say it that way. So prophase 1 is about forming tetrads. And here's a spindle here connecting there, and here's a spindle there connecting here, and here's one to there, and here's one to here, and here's one to here, and here's one to there. So our goal in prophase 1 was to form the tetrads.
What do spindles connect to?
Kinetic course, just like before. Now, prophase is where we made the tetrads. Metaphase is where we're going to move the tetrads. Just like in mitosis, we break down and or build up our spindles to move the tetrads to the metaphase plate. So at the end of this process, homologous chromosomes 1 are lined up on the metaphase plate. Chromosomes 2 lined up on the metaphase plate. Chromosome 3 tetrad lined up on the metaphase plate. Centrioles, spindles. All right, so far so good. Just gets worse from here. So if this doesn't make sense, let me know now. My chair is low. I seem to be sinking down out of the picture. All right, excellent. Okay, perfect. Now notice something, the way these lined up. And I did this on purpose. Notice for chromosome 1, mom's chromosome is on the top of the screen. Whereas for chromosome 2, mom's chromosome is on the bottom, and chromosome 3, mom's is on the bottom. What determines which way they line up, whether mom's on top or dad's on top? What do you think determines it? Exactly, it's random. It's like flipping a coin. And when you flip a coin, what are your chances of getting heads?
50-50.
50-50, right? A 50-50 shot. If you flip 2 coins, what are your chances of getting 2 heads? %25. How do you know that? Yeah, half on one, half times the other, exactly. In fact, that's how you would calculate this. It's basically 1. When you're doing this, if we were to do the math of this, you would take 1, and that 1 would basically be over whatever the number of options there would be. In this case, it would be 2. Where's my, to the second power, all right? 2 different times you're flipping the coin, and each coin has 2 options. That would equal 1/4 or 25%. Now, how many pairs of chromosomes do we have? 23. So that's like flipping 23 coins. What is 2 to the 23rd power? Come on, you're all on computers. You all have smart devices. I will sit here until someone gives me the number. No, we're not getting away with just saying really big. I want the number.
529.
Nope.
Oh, 20.
2 to the 23rd power.
Oh, sorry.
There you go. 8.4 million different possible combinations. When your 23 chromosomes line up, the combination of how they show up, there is almost 8.5 million different ways that these can line up. We call this independent assortment. Remember, oh, why didn't that write? One of the reasons why you look similar to your siblings and not identical to them is because when mom was making her eggs, her independent assortment gave her over almost 8.5 million different possibilities. Then dad's got 8.5 million different possibilities when putting his together. So while you may look similar to your siblings, is anybody identical to their siblings who's not an identical sibling? No, absolutely not. And that whole Petty Duke thing is total BS. And I know none of you get that reference, and I don't care. That is one of the jokes I tell solely for me, and I don't care that none of you get it. It makes me happy. All right, excellent. The point being that there is an absolutely massive number of combinations that we can put together of just how we randomly assort this. And as we'll see, there's even more that goes into it as well, but we're not there yet. All right. Prophase, we made the tetrads. Metaphase, we lined up the tetrads. Guess what we do in anaphase?
Separate them.
Exactly. Split the triads. Tetrads, sorry. Split the tetrads, and I want that to be black again. Just like in mitosis, in this case, our synapsis inactivates. And when that synapsis inactivates, then our homologous chromosomes are separated. And they are pulled to the opposite sides of the cell. And we still get that cool V shape as a result of it. So this X is pulled this way and has that cool V shape. This one gets pulled this way and still has that cool V shape. Dark green V's going this way. Light green V's going this way. Dark blue going this way. Light blue going this way. Excellent. And so we split our tetrads. And so then here in telophase, and we should also say, here, let's do this. Telophase 1 and cytokinesis 1. we get squeezing of the cell, where that cleavage furrow forms as the actin ring pulls along the metaphase plate, separating it into two individual cells. Oops. And again, nuclear envelopes are going to reform. Our loose chromatin condenses down into the dense chromosome. And we have the loose chromatin of our cells now floating around inside of here. And notice, in each case, how many chromosomes does each of these nuclei have?
3.
3. So this one has 3 chromosomes. And this one has 3 chromosomes. But notice the chromosomes are still replicated. And so again, that is why we now refer to this one. Well, this one up here was 2NR. This one is now just NR. This one is just NR. So we went from a, well, this one is actually just 2N, sorry. Our original drawing was 2N. But we went from a cell that was 2NR, diploid and replicated, to haploid and replicated. We now have haploid, replicated chromosomes. Number of chromosomes inside these cells at the end of meiosis one. So notice meiosis one is all about tetrads, forming them, moving them, splitting them, and then really unwinding the tetrads.
May I ask?
Absolutely.
So in this meiosis one, we just separated women and males chromosomes from each other.
I'm sorry, say that again.
In this meiosis one, we separated women and males chromosomes from each other, right?
So yes, so you are correct. So, okay, great. So that takes us to the second part of what we're talking about. Yes. So notice in this instance, the chromosome that came from mom goes one way, and the chromosome, the homologous chromosome that came from dad goes the other way. So notice if this was our organism and it only had 3 chromosomes, each one of these cells has a copy of chromosome 1, a copy of chromosome 2, and a copy of chromosome 3. Now you are right. The way we've done this right now, this top cell got all of mom's chromosome 1, and dad got all, the bottom one got all of dad's chromosome 1. And is that how it works with your kids? When you have a kid, does that kid have both your ears from your mother and also your eyes from your other, and also your teeth and the hair from your mother, and they get nothing from grandpa?
No.
No. And that's because there's another way that we improve the randomness of this process. Remember, independent assortment, just how they line up, give us over 8 million different variations in genes that we can give our offspring. But there's another important process that occurs here during metaphase, well, between prophase 1 and metaphase 1. During this time, the second process that occurs is this process called crossover. In crossover, what happens is portions of one homologous chromosome. Need more room. Portions of one homologous chromosome trade places with the other homologous chromosome. So while these tetrads are forming, while they're attaching to each other, what will happen is that, for instance, a piece of moms will break off and attach to dads. And a piece of dads will then replace that same part on moms. So then notice now, when they separate, this cell isn't all moms. It has a piece of moms, one that's all moms, but one that's part mom and part dad. This one has a little bit there that it mixed off with. This one's got a little bit there. And then this one's got a little bit there. So notice with just one crossover, I now have even more variety, more variations. And is that crossover always going to occur in the same place every time? No. Dozens of crossovers can occur back and forth and back and forth and back and forth, basically giving us an almost unlimited number of variations that we can form when we produce our gametes. But for simplicity, we will just stick with that one crossover to see what happens at the end of this process. But remember that not only do we have that independent assortment that's giving us variety, but also we have this crossover that's going to give us limitless variations in how we jumble together our genetic information, giving us these incredibly unusual combinations of characteristics that we call our offspring. All right, where they've got grandma's ears and grandpa's stubbornness. Right? Although what's really weird in my family is it's turned out all the bad traits came from my wife's side and all the good traits came from mine. So again, sometimes some unique things happen that way as well.
Funny.
All right, questions on that? All right, excellent. Well, we're only halfway through meiosis. However, the good news is that meiosis 2 is basically identical to mitosis. So let me save this one more time and so I can clear it, which I really don't want to do because I kind of wanted to keep all this stuff up at the top. But okay, so actually let's do that. Let's make this and make it white so that I can do that. Oops. Put that there. Put that there. There. Excellent. Oops, missed you. Perfect. All right, great. So now what we can do is meiosis 2. Meiosis 2, just like mitosis, it's all about arranging and dividing replicated chromosomes. So if you think about it, we start with cells, 2 cells that are going to be NR, haploid and replicated. And we are going to end with 4 cells that are just N, haploid. All right. So not surprisingly, again, we have prophase, metaphase, anaphase, and telophase. Prophase 2. Metaphase 2. Phase 2. And telophase 2. So again, prophase 2, our centrioles migrate to the poles and start making their spindles. Our nuclear envelope breaks down, and we get the formation of our chromosomes. And remember, this first cell had chromosome 1 from mom, chromosome 2 from dad, chromosome 3 from dad, but remember it had a little piece of mom on it as well. Metaphase is when we line these up on the metaphase plate. So again, my chromosome lines up. My chromosome lines up, each of these independently. And my chromosome lines up. And again, on one side, it's going to have that little bit of moms on dads. Anaphase, the centromeres inactivate. The chromosomes get pulled to opposite poles. And in telophase 2 and cytokinesis, our one cell is divided into two cells. Our nuclear envelopes reform. And we have two haploid cells with half of the number of chromosomes, making them uniquely different from the original. But notice also, thanks to our one singular crossover, they are also uniquely different from each other. So here we have two unique cells, haploid cells. And of course, the exact same thing, happened on the other cell as well. So we got that other cell, bang, bang, boom. And for simplicity, this time I'll just draw the chromosomes. So this one, chromosome 1 lined up, chromosome 1 got pulled apart. And to make more room, let's do this here. Oops, no. Two, chromosome, oops, no. So it needs to be lined up on the metaphase plate, pulled to the opposite sides. Oh, this is chromosome 3, oops, which means I need to put that there. I need to put that there. I need to put that there. And I need to put that there. And then I can do chromosome 2. Two unique haploid cells. And so total, we have 4 cells with half the number of chromosomes. Each has a copy of chromosome 1, each a copy of chromosome 2, each a copy of chromosome 3. But because of crossover, every single one is unique and different from the other. And notice it only took one crossover to make them each unique to each other. And dozens of crossovers occur during prophase 1. Again, this should hopefully mostly be review because the meiosis that you have done. But I wanted to make sure we went over it together. And again, to truly beat the dead horse, we have the great pictures from your textbook that show this as well. We here see the difference between mitosis and meiosis. And conveniently enough, they used a cell like mine that has 3 unique chromosomes, 6 total, so 3 pairs of chromosomes. Again, mitosis, we replicate them, we separate them independently, we end up with 2 cells identical to each other and identical to the original. But in meiosis, we have 2 divisions. And Meiosis One is about forming and dividing tetrads. So we form a tetrad and divide the tetrad. Form a tetrad and divide a tetrad. Form a tetrad and divide a tetrad. That way we are certain that each of the first cells that are produced get 1 copy of all of the genetic material. And then Meiosis 2 is about dividing the chromosomes. 1 replicated copy to one side, 1 to the other. Now notice without crossover, these 2 cells are identical. These 2 cells are identical. They're different from each other, so 1 is different from 3 and 4, and they're all different from the original. But notice by just having that 1 crossover, we get 4 unique cells. Here, we see that crossover occurring. So notice here we're getting some crossover that is occurring, giving us unique cells. Notice this one has 2 crossovers that occur. They divide. And at the end of this process, notice they only showed the second prophase for just 1 set of cells. But here we have the 1 cells and the other 2 would be identical as well. So again, hopefully this should be pretty straightforward because you guys got this in the Labster and then we went over it here. So any questions on this? Oh, one more pretty picture. Oh, and this one is from the other ones weren't from your textbook. This one's the one from your textbook, which I like because it shows crossover. But the other thing that it shows is it shows both divisions of the cells. So again, notice at the end of this process, 4 unique cells with half the number of chromosomes. And thanks to crossover, each one is unique. All right. So if that all makes sense, then the good news is we're really 2/3 of the way through spermatogenesis. Because remember, phase 1 is mitosis, and we understand that. Phase 2 is meiosis, and we understand that. So now all we have to do is 2 things. We have to do spermiogenesis, and because mitosis and meiosis occur in both males and females, we have to add labels to all of these cells. All these cells at different stages in this process have to have unique names so we can talk about how this specifically happens in males. And then when we get to females, we will specifically talk about these processes in females, the timing of it, the names of the cells, and we'll talk about the differences. And being the smart, sophisticated students that you are, not only are we going to talk about spermatogenesis, we're also going to talk about oogenesis. So when we have 2 related processes, how many possible essay questions can you have? 3. Describe spermatogenesis, describe oogenesis, and what's the third? Compare the 2, absolutely. How are they similar? How are they different? Excellent, 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 excellent. So that is what we need to do next. Now that we understand mitosis and meiosis, we don't have to worry about talking about each of the individual stages. We can talk about what cells we start with and what cells we end with in these processes. And we can now talk specifically about this gamete formation in males. All right, now we've covered a lot of material and we got a lot more vocabulary coming as well. So this is another good natural stopping point. We've been talking for almost exactly 1 hour. So it's 2:40, so let's come back at 2:55. And at 2:55, we will restart. And I will start the recording at that point. So any questions on the generic process of mitosis and meiosis before we take our break? All right, then I'll see you in 15 minutes. All righty. So we've talked about mitosis and meiosis, and now we're ready to talk about spermatogenesis. Any questions? All right. As I have mentioned, as we look at the seminiferous tubule, as these processes of mitosis and meiosis and spermiogenesis are occurring, we are going to be migrating from the basal surface of it, where the myeloid cells are, towards the lumen. And so we will actually be able to identify these different stages and therefore the cells in these different stages as we work our way towards the lumen of our seminiferous tubule. So again, we start with, at the basal surface, our stem cell, and that stem cell is a spermatogonium. Those are going to undergo mitosis, and then we are going to go through the immature sperm, including our primary spermatocytes, our secondary spermatocytes, and then finally our immature sperm-shaped cells, which are our spermatids. So these are going to be the stages that we're going to see as we go through this. So let's take a look at this. And then ultimately, our goal is to make that mature spermatozoon, or spermatozoa for the plural. So up here at the basal surface of our seminiferous tubule, we have our spermatogonium, our stem cell, that undergoes mitosis. Oops, there we go. Now, oh, and I think we mentioned this earlier, but again, let's go ahead and start with that. Spermatogenesis begins when? When did we say spermatogenesis begins? Puberty, excellent. At puberty, and when does it end? What did we decide? 15 minutes after death? Oh, that was a good line. So we'll go with that, 15 minutes after death, right? It is pretty much a continuous process from puberty till death for males. So again, we've talked about how we need to make a gajillion cells. So this process starts with our spermatogonium undergoing mitosis. Again, we are going to get one cell, that's spermatogonium, that is going to form two identical cells. These are going to be the type A and type B cells. Let's do it this way. And as we talked about, what hormone drives this process? Excellent. And since we've written it out once before this lecture, I'll go ahead and abbreviate it here as well. Follicle stimulating hormone. Follicle stimulating hormone is what regulates this process. Now, with these two cells, again, they're identical cells, type A and type B, but while they're identical cells, they are going to have different ultimate destinations. The type A cells stay at the basal surface, and basically they become new spermatogonium. Basically, that type 1 cell replaces the first cell that divided. So a cell divides, and now we have a cell to replace it. This is why this process can continue infinitely, because the first thing we do during this process is make a cell to replace the original one that divided. However, notice the type 2 cell, type B cell, starts migrating inward. And as it migrates inward, it undergoes a change. And it prepares to undergo meiosis. And this cell, this type B cell, becomes our primary spermatocyte. So our primary spermatocyte is that newly divided type B cell that is getting ready to undergo meiosis. And that's what we see here nicely in this illustration. Here in this illustration, our spermatogonium undergoes mitosis, forming a type A cell that stays a spermatogonium, and the type B that starts migrating inward and becomes our primary spermatocyte. Now, this primary spermatocyte undergoes Meiosis One, formation, lining up and dividing of the tetrads. And after that first division, we get 2 haploid cells with replicated DNA. Again, notice here they've just got them labeled as N, but remember we know that they're haploid and replicated. So really they should be NR. Right? Otherwise you wouldn't be able to go from N to N. You'd go from N to half N or something like that. So the way we're able to go from N to N is because these are 2 haploid replicated cells. Guess we should write that. Well, let's say it this way, 2 haploid cells with replicated DNA. I like that better. And these 2 haploid cells with replicated DNA, we call our secondary spermatocytes. Which again makes it convenient. How many primary spermatocytes do we have? Primary is first, and we have 1. Secondary is second, and how many secondary spermatocytes do we have? 2. That makes it easy to remember. These secondary spermatocytes then undergo meiosis, undergo meiosis 2, and they form our early spermatids. These are our 4 unique haploid cells. But more specifically, there are 4 unique cell-shaped haploid cells. And then these early spermatids undergo spermiogenesis, the shedding of most of their cytoplasm, until they become late spermatids. And it's these late spermatids that finally reach the lumen of the tubule and are moved out of the tubule to the epididymis. There we go. And it is finally in the epididymis where they mature with our androgens. into our mature spermatozoa. Now, your book actually does a really nice job of describing the process of spermatogenesis. Because we only have 2 weeks to cover this material, I'm not going to hold you responsible for all of the steps involved in the process of spermiogenesis, but we do need to know what occurs during this phase. Again, we are going from a cell-shaped cell to a sperm-shaped cell that has 3 distinct regions to it that we will identify and describe in a moment. But as you can see, basically it is shedding most of its cytoplasm and specializing the regions for motility. Again, those nerve cells release the chemical stimulants that stimulate this process, and they phagocytose the excess cytoplasm in this. So they're driving it and recycling the components. And when we look at a mature sperm, and we'll talk about the anatomy of this in a second, it takes us about 70 days to get from stem cell to 4 sperm-shaped cells, right? And remember, even at that point, it's not viable yet. It has to go to the epididymis, where it takes 20 days for it to mature. So from stem cell to mature sperm that is capable of fertilizing an egg takes about 90 days, right? And you thought the lines at Disneyland was long. Just imagine waiting 90 days for a 3-minute ride. All right? Questions on that? All right, excellent. Now, as I mentioned, when we look at the anatomy of the sperm, there are 3 distinct regions to it. The first is the head, what is also known as the genetic region. This is what contains the DNA. As I mentioned, this is basically a DNA missile whose job is to get to the egg and fertilize it. Now, while the majority, and this picture doesn't do the best job of showing it, while the majority of it is the nucleus that contains, and so where's my highlighter? Is the nucleus that contains the genetic material. There is a large cap-like structure on the outer surface of this, known as the acrosomial cap. Again, for those of you who have taken me for 430 and for now 3/4 of 431, you know that I commonly refer to the outer layer of something as its candy-coated shell. Well, as I mentioned when we were talking about the endocrine system, the ovulated egg that is released by the female actually does have a glycogen-rich protective coating on its outer surface. It really does have a candy-coated shell. So to be able to penetrate those defenses so that the sperm can get inside, this acrosomial cap contains a large amount of digestive enzymes. So what'll happen is when it gets to the egg, it will release those digestive enzymes, and then it will powerfully whip its tail, digging its head into that glycogen-rich layer to try to dig away at that candy-coated shell, penetrate that candy-coated shell, and get in and fertilize that egg. And that acrosomial cap plays a huge role in that. The mid piece of this is also what is known as the metabolic region. This is where that motor that I mentioned is located. That motor, like I said, like a gear, has a ratchet that rotates the tail or flagellum around. And as I mentioned, every notch of that gear requires an ATP to just move that little bit. So if I'm going to whip that tail around, I need a massive amount of ATP. And what you don't see as well in this picture, although I think the previous picture actually showed it even nicer. The mitochondria, massive numbers of mitochondria wrap around the mid piece, wrap around that motor. So right there where the motor is located is where we're going to have massive numbers of mitochondria. Yeah, like a drill bit. Massive amounts of mitochondria to be able to produce all the ATP necessary for that swimming motion. But it is a massively exhausting process. So like we said, while these things are sitting around for their 90 days to mature and be released, they don't want to be sitting there spinning their tails. They would be wasting so much energy and so many resources and exhausting the cell in that process. So that's why it relies on peristalsis and fluid pressures to move it through the ductwork of the male. Lastly, as we mentioned, we have the locomotion region of the tail, which again is that specialized organelle, the flagellum. And again, it gets rotated in a whip-like fashion to propel this cell, being the only cell that is capable of propelling itself through space by itself. All right. Questions on that? All right, let's look quickly at an illustration of our seminiferous tubules so we can again see the anatomy of this here. And as we do this, we can then highlight the different cells. Remember, as we talked about, on the basal surface is where we are going to find the spermatogonia, the stem cells that produce all of the sperm are located on the basal surface. The primary spermatocyte is pretty easy to identify because it is going to be the largest cell. If you think about it, this is the cell that has to divide twice to produce 4 cells. So that means it leaves a lot of stuff inside of it. So it is by far the largest cell that you are going to see inside the seminiferous tubules. And notice it's not on the border. It's away from the border. A little bit away from the border is where you're going to see that big, huge, massive primary spermatocytes. When it divides in half, it forms the much, much smaller secondary spermatocytes. And then the cell-shaped cells that are closest to the lumen are going to be the early spermatids. Early spermatids are the cell-shaped haploid cells, whereas late spermatids are the sperm-shaped haploid cells. And then, like we said, when they mature in the epididymis, they become mature spermatozoa. Now, it's easy to see this in the pretty illustration, but I am pretty sure, nope, I lied. So let's do this. Let's actually look at it histologically. Got a bunch of these pictures. Let's, I like this one. Let's go with this one. So again, here we see, these arrows are not mine. This is just a picture I found on the interwebs. But notice as we look at this, again, as we go from the surface where the myeloid cells are located towards the center, we can see the different cells. So notice first, The ones that are closest to the surface, closest to the basal surface, are the Spermatogonium. Notice much, much bigger than that, the largest cells, so they're very easy to find. The largest cells that we're going to see are going to be those primary spermatocytes. The primary spermatocytes are going to divide into the much smaller secondary spermatocytes. So here's a good place where we can see this. We can see these secondary spermatocytes that are much smaller than the primary one. And then notice closest to the lumen are going to be the early spermatids, followed by these where most of the cytoplasm has been released. And these are examples of late spermatids. And notice you can see the flagella as they are coming out into, giving the lumen this kind of dirty appearance from all of those flagella as it goes towards the center. Hold on, there is a question. Great question. They're really, no, so they're not going to be released. The cytoplasmic bridge that kind of forms in between them, just because this process is such a rapid process, you don't get while it's dividing, there is still going to be some of the cytokinesis that's going to be occurring. I wouldn't say that it probably forms any major function. I mean, I guess the one thing you could think is possibly it helps to allow them to share the cytoplasm, but remember, they're going to be expelling most of the cytoplasm. So I don't really think that there is a significant reason for why they don't fully divide. I think it's more of just having to do with the speed of this process. Again, remember, why is one of those questions it's always hard to answer. We can kind of infer why some of those whys might be. I don't know if we necessarily have the reason for that. I wouldn't say that there's necessarily a meaningful reason for it. Because again, it's not like we have to keep track of them once they're divided. It doesn't matter that way. So it's more just probably because they're due to the speed. All right, good question though. Any others? All right, excellent. So we looked at the nice illustration, and then also looking at a seminiferous tubule histologically, we can identify these cells pretty easily and distinctly as well. All right, so we're back here. All right. So far, are you guys seeing the lecture? I can't tell.
Yes.
Okay, perfect. So far, everything that we've been talking about happening, remember, is all happening here in our seminiferous tubules. All right, so this is where we are going from stem cell to sperm-shaped cells, those late spermatids. But as late spermatids, it is now time to leave our seminiferous tubules and get into that epididymis for maturation. To do that, we have to take a rather, not necessarily precarious, but elaborate path. Notice to leave each individual lobule, we go out of a structure that is known as the straight tubule. That straight tubule feeds into this mesh or net-like structure known as the rete testis. The rete testis then feeds into the tubule structures that actually leave the testis, and because it comes out of the testis, not surprisingly, it is the efferent ductule. So again, if we're thinking of the path of this closed duct system, goes from the seminiferous tubule through the straight tubule into the rete testis. And again, they're not spinning their tails to do this. It is the fluid pressures from the fluid that the nurse cells are making and from the contractions of those myoid cells. The rete testis collects the immature sperm from all of the lobules and feeds it into the epididymis, again through that efferent ductule. And it is in the epididymis where we are going to process the sperm, maintain the sperm, but also mature the sperm. The epididymis is a relatively large comma-shaped, tightly coiled structure, a little over 20 feet in length if you were to take it out and stretch it out. It is associated with the testis, connected to the testis, but not a part of the testis. While you are observing that male's Raffi, while you're having him contract his cremaster muscle for you, also ask if you could palpate the epididymis. And what you will find, if again this is, we'll say it is your testis, your left testis, then basically where the epididymis would be is superior, posterior, and lateral. So basically there's this comma-shaped structure that as you palpate that oval testis, you'll feel this bump along the superior, posterior, and lateral aspect. This comma-shaped structure is this long coiled tube known as the epididymis. The epididymis has three regions: an enlarged head, a body, and a tail. The composition and the fluid of them are slightly different. What happens in them is slightly different. We're not going to worry about what happens in each individual portion of it. We'll just talk about what happens in general. And in general, the epididymis has basically three main functions. The first is that it needs to maintain the composition of that fluid. Those nurse cells were making the fluid and sending the fluid into here. And this fluid is going to have the nutrients, it's going to have the oxygen, it's going to have the removal of waste to keep these millions of sperm alive. Here in our epididymis, we're going to run into a very, very interesting tissue. A unique tissue that this is the only place in a human's body where we will find it. And that is a non-motile ciliated pseudostratified columnar epithelial tissue. Normally, when we think of a ciliated pseudostratified columnar epithelial tissue, we know those cilia are motile for the movement of materials. And in fact, if you think about it, that would make a heck of a lot of sense in here. If we didn't want the sperm to move themselves, then why not have cilia that could move them for us? Again, why is one of those things we can't easily answer. But my guess is that it's probably the cilia would move them too quickly. Remember, it's a little over 20 feet long, but the sperm need to stay in here for 20 days. My guess is that cilia probably move at too fast of a rate where it would move the sperm through too quickly. But again, why they're not motile, I don't know exactly, but this is one place, the one place in the body that you will find a ciliated pseudostratified columnar epithelial tissue that is not motile. Instead, those cilia just give us big, long extensions that increase the surface area for the absorption and secretions of substances into the fluid. It is going to store and mature the sperm. And remember, to do that, it is going to need the help of those androgens. And those androgens, remember, came from the interstitial cells and were carried by the androgen binding proteins that were produced by the nurse cells. So those androgen binding proteins are chock filled with androgens. So the fluid in here is chock filled with androgens so that we have those androgens. And I think I'm going to need to sneak this up here and maybe make it smaller so it fits. And again, it's not a fast process. It takes about 20 days to mature these sperm. And lastly, and this gets back to that quality control we were talking about before. Here in the epididymis is where we are going to be able to monitor the condition of the sperm. If there are abnormal sperm that are abnormal chromosomally, so it could be chromosomal abnormals, incorrect divisions. Or it could actually be morphological abnormalities. Like having two heads, or one head with two tails, or a tail that is too short, or a tail that is too long, right? Characteristics like that, those cells can be weeded out here in the epididymis and be removed. Now, again, as we were talking about, when we're talking about millions and millions of cells, it's not going to be a perfect system. And a good number is somewhere around 70. If we can get 70% of the sperm to be viable, then that's going to be useful. And again, not all abnormalities are going to be something that are going to be indicated. While it can catch some chromosomal abnormalities, can some get through, leading to chromosomal disorders like Down syndrome and things along those lines? Absolutely. But this process also takes time as well, right? Remember, one of the things we talked about is that the more time it has here in the epididymis, the better the recycling, the better the filtering, the better the processing and the quality control is going to be. And while you probably haven't thought of it in those terms, most of you, especially those of you who have ever tried to get pregnant before, are aware of it. Because if you and your significant other are trying to get pregnant, the week of your ovulation, do you have sex 16 times a day for all 7 days of that week, just hoping to catch it at the right time to get pregnant? No, because when you're going through the sperm that quickly, they have to be processed more quickly. And if they're processed more quickly, there's going to be more errors, and there's going to be less processing, less quality control of it as well. And the number of viable sperm with multiple ejaculations can actually drop below 50%. And as we'll see, to effectively fertilize the egg, we need a massive number of viable sperms. There is a massive amount of attrition that takes place, mostly because none of them will stop to ask for directions. All right. So again, instead, you try to get more focal on when that ovulation is going to be. You don't have any intercourse for 2 or 3 days before that to try to maximize the viability of the ejaculate that you produce. All right. Questions on that? Yes. In general, the male should ejaculate less, and especially avoid ejaculating, should ejaculate less in times outside of ovulation. Again, 2 or 3 days, right? Again, just because you're going to be ovulating 3 weeks from now doesn't mean that you should say no. Although if you want to use that as an excuse, feel free to say that I said that. But no, usually at least 2 to 3 days prior to when you anticipate ovulation to occur. And then usually they not more than once or twice a day for the 2 or 3 days around there, again, to try to maximize the viability and the volume, the number of sperm that are going to be produced with an ejaculate. Yep. All right. Excellent. Once matured, they're going to move through the ductus deferens, or the vas deferens, both of those terms are acceptable, into the abdominal pelvic cavity, or more specifically through the spermatocord back into the abdominal pelvic cavity. And are the sperm going to be swimming through the ductus deferens? No. In this case, the ductus deferens will primarily be using peristalsis, those rhythmic contractions of the smooth muscle to move the sperm through the ductus deferens into the abdominal pelvic cavity. Notice here the ductus deferens comes up through the spermatocord and over the pubic symphysis. Someone remind me again what the pubic symphysis is? What is the pubic symphysis? Excellent. It is a fibrocartilaginous joint. Helps to form the pelvis where the two pubic bones come together. A little bit of fibrocartilage forms that pubic symphysis. To enter into the abdominal pelvic cavity, what happens is the ductus deferens comes up basically the pubic bone right next to the pubic symphysis and in that inguinal canal. Again, while you're observing the raphe and the contractions of the cremator muscle and palpating the epididymis, you should also gently palpate the pubic bone for the male, find the pubic symphysis and go a little bit lateral from there. And you can actually feel, you should be able to feel the ductus deferens. You should actually be able to palpate it against the bone of the pubis and actually be able to feel that ductus deferens in there. Again, you want to be careful because remember, blood vessels are going through there as well. So don't just mash it with your hand. You want to gently palpate it, but you can actually feel the ductus deferens as it works its way through. Notice the ductus deferens goes over the top of the bladder, hooks around the ureters coming down from the kidney, and then has a big, huge enlargement of it known as the ampulla. This ampulla is where we store massive amount of sperm in anticipation of release during ejaculation. And how long can the spermatozoa stay in the ampulla of the ductus deferens? Does it fill up and require releasing? Otherwise, the male is in tremendous pain and could die as a result of a lack of release from all of that buildup of sperm in the ampulla. Yeah, many guys use that excuse, but in reality, it can last for over a month. The sperm can last for well over a month in that ampulla of the ductus deferens. But as long as whoever you're doing that with doesn't listen to this video, then you can still use that excuse, Alex. All righty. Yeah, here, show her this part. Yes, lack of release of the spermatozoa can actually potentially cause a man to die. All right, there you go. Excellent. All right, questions on that? All right. As we mentioned, it has to pass through that inguinal canal into the pelvic cavity. This is useful because being an outside structure, outside the body, over that pubic bone, and again, can be stored for months in there. Moved via peristalsis, as we mentioned. The location of the spermatocord and the ductus deferens makes it very easy for the male form of birth control. What is the male form of birth control? I mean, other than saying no. A vasectomy, absolutely. And with a vasectomy, that vasectomy is a cutting of the spermatocord. Basically, at the base of the scrotum, right underneath the root of the penis, they make an incision. Sometimes they'll make two, one for each side, but often the doctor will make a singular midline incision. And what they will do is they will go in and pull out the spermatocord. Now, when they first started doing this, what would happen here? Let's cheat. I think I saved this, but I don't remember. With that cord, they would pull the cord out, and what they would do is they would actually take a clamp. And they would take a clamp and place it over the tube, clamping it on the tube, and then putting the tube back inside. Why would they do something like that? Why put a clamp on the ductus deferens? Yeah, exactly. The advantage is that it was reversible, right? If something truly tragic happened, like your kids died in an accident or your wife became unattractive, you would have the opportunity to reverse this and have children with somebody else, right? The problem with that is the clamps didn't work very well. So instead, what they would do is they would just go in and they would cut it. What's the problem with that? True, it's not reversible, and exactly, well, I guess that is a problem. In fact, before you can have a vasectomy, especially nowadays, since most people are doing it through HMOs, there are actually courses you have to take. I made the joke about, being able to reverse it and everything, but you really do have to go in, and they have a course where you have to make sure you understand when they do this surgery is expected to be permanent. So if your kids do die in a horrible accident, you're toast. They encourage you to bank your sperm if you think there's an outside chance of somewhere along the way that you would want to have kids again. But for the most part, this surgery is not reversible. The problem with just cutting it is that sometimes it would grow back together. Exactly. The ends would come back together. So now what they actually do is they now go in, pull out the cord. They will actually cut out a section of that cord. They then go in and cauterize the ends to basically eliminate essentially any chance of it regrowing and reconnecting back together. Now, this is an incredibly non-invasive surgery. It's a weekend surgery, the kind of thing that you can have on Friday and be back at work on Monday. It is done outpatient. One of my favorite things all time, and I don't know if they do it anymore, but there was a urologist in, I think it was Wisconsin or something like that, when their basketball team was really good, that they had a March Madness special. They figured you were going to be sitting on the couch for the next 3 days watching basketball anyway. So you got a discount on your vasectomy, a discount on a big screen TV, and a free pizza. So that you could sit there in the chair with a bag of frozen peas on your testicles, bringing down the swelling, watch basketball for 3 days, and then be back at work on Monday. Now, in some rare instances, there can be swelling, there can be some pain and discomfort that comes from this vasectomy, in which case, and again, especially because we're talking about HMOs now, where they will try to go back in and reconnect it. But they can't guarantee that. So they can try, they will sometimes try to reconnect it together again, but there's no guarantee that scar tissue won't form and stop the sperm from being able to get through. And what they found is about 2 years out from a vasectomy, the testes actually stop producing sperm, or at least the production of it decreases dramatically. So what they find is if 2, 3, 5 years later, you go back and try to have your vasectomy reversed, often that male isn't going to be able to produce enough sperm to be able to reliably be able to fertilize someone and have kids anyway. So at this point now, that vasectomy really is a one-way journey. Great question. No, ejaculation will still occur. And let's actually go back to the picture. Is a great question. Hold on. Let's there. Do I want to be here? Yeah, this will work. Yes. The answer to that is yes, because the ejaculate that is produced by the male is not just sperm. Yes, we have the ductus deferens and its ampulla that is filled with sperm ready to go. But sperm is also a mix of, ejaculate is a mix of sperm and fluids. Our glands, the seminiferous tubules, the prostate, the bulbourethral glands. These produce the fluid component of the ejaculate. In fact, in a normal male, sperm only makes up about 5% of the ejaculate. So after a vasectomy, when there's no more sperm, you only see a minor decrease, and for most people, it's not noticeable. So yeah, the ejaculate won't have a sperm in it anymore, but you'll still produce an ejaculate because there will be lots of the fluids still being produced as well. Because remember, the cut is going on outside the body. All right. Now, females. Can we do a birth control in females as well? And again, not the pills, but surgically. Yeah, what do we call that? Yeah, getting your tubes tied, exactly. However, that procedure, that procedure is a, it is not an outpatient. It is an inpatient surgery. Recovery can take 2 to 3 days from this. It requires abdominal pelvic surgery to take place. It is a much more serious, much more risky process in females than it is in males. So typically for most couples, it is much safer, much smarter for if they don't want to have any more kids, for the male to have the vasectomy. No, the tubal ligation is not a process that is typically considered to be reversible. All righty. So again, as we talked about, we have those ductus deferens or vas deferens. Notice it's a posterior view. So it comes up over the ureters of the bladder into that ampulla. And then notice it connects to the duct of our seminal vesicle or the seminal gland. And they form a structure known as the ejaculatory duct. And the ejaculatory duct feeds into the urethra. So again, if you think about it, remember, we said in males, this is a closed duct system. From start to finish, we start in the seminiferous tubules. From there, we go into the straight tubule. From there, we go into the rete testis. From there, we go into the efferent tubule, efferent ductule, into the epididymis, into the ductus deferens, into the ejaculatory duct, into, and if we want to be more specific, prosthetic urethra, into the membranous, into the spongy urethra, and then out.
You skip the ampulla of the ductus.
True, you're right. The ductus deferens at the end does have an enlargement called the ampulla, but the ampulla is not an additional structure. It's not an additional tube, but I guess you're right. Since we talked about the parts of the urethra, it doesn't hurt to talk about the parts of the ductus deferens. So we would do the ductus deferens into the ampulla. And I guess for that matter, even though we don't need to distinguish them, we would go through the head, body, and tail of the epididymis too. So I guess we could do that as well. Yes, Ashley, I would say the same thing. Males tend to be worried about getting the procedure done. But like I said, in comparison to the risks associated with female birth control versus the male birth control surgeries, I hate making these types of broad statements, but quite frankly, it's selfish of the male to put that risk on. If you're in a serious monogamous relationship, then the risks associated with it's a major surgery in females, and the risks associated with that compared to the outpatient procedure that is a vasectomy seems pretty irresponsible to put somebody you care about at that serious risk for something like that. So again. All righty. Either that, or maybe it's just the female nurses seeing their junk when they're doing the cut in there. I don't know what makes them so scared. All righty. Any other questions on the anatomy of this? Maybe. All right, excellent. And again, as we know, the urethra extends from the base of the urinary bladder to the tip of the penis. The entire urethra is lined with a transitional epithelial tissue, with the exception, as we've talked about before, about the very distal part. The very distal part, remember, is going to be lined with that non-keratinized stratified squamous epithelial tissue, where it can be, because it's, again, exposed to the outside world, but the rest of it is going to be a pathway for both urine and semen. So lined with that transitional epithelial tissue. And as we talked about, has those 3 distinct regions to it. All right. Questions on that? We are mostly done. We have a little bit more that I wanted to cover because, again, I want to finish the rest of the anatomy for the male. So let's do this. Rather than taking a break now and then going all the way, I don't think, hold on, let me see what we have left. Yeah, I only have a handful of slides that I wanted to go through to finish this. So why don't we go ahead and do that, and then we can finish early. So rather than taking a break and going a little later, let's push through and go ahead and just finish for today. All right. We have talked about the testis. We have talked about the ductwork system. What we are left with to talk about then are the accessory glands. So let's talk about those. The first, as I mentioned, are the paired seminal vesicles, or also known as the seminal glands, the singular prostate, and the paired bulbourethral glands. And as I mentioned, they produce about the majority of the ejaculate. The spermatozoa and the fluid from the nerve cells and the epididymis only make up about 5% of the semen, of the ejaculate. The rest is made by those 5 major glands. So what is the function of these? Well, again, those sperm have been sitting there cooling their jets, waiting for the opportunity. So it's time to flip the switch and start that spinning motion. We need to activate the spermatozoa so that they are able to propel themselves to the egg. That spinning motion requires a tremendous amount of energy. So as such, we need lots of nutrients, lots of fuel for those spermatozoa so that they can do what they need to do. But we do also want to help them. Ejaculation in the male involves peristaltic contractions to help in the propulsion of the sperm. But we also want peristaltic contractions in the female as well. If we can stimulate the female's reproductive tract to undergo peristaltic contractions, that can help to draw the sperm up to where the egg is located. And there are two ways we can do that. One is by inducing orgasm. When you induce an orgasm in a female, that causes the female reproductive tract to undergo a reverse peristalsis that can draw the sperm up to the egg more efficiently. But also there are chemicals, hormones and hormone-like chemicals in the semen that is going to stimulate peristaltic contractions in the reproductive tract of the female as well. This is one of the reasons why for individuals who have been pregnant before, one of the things that the doctors likely warned you about is having intercourse late stage in the pregnancy, because there could be a concern that those chemicals in the ejaculate or an orgasm that is produced can actually inappropriately kick off that positive feedback process that leads to parturition, expulsion of the baby. So it can inappropriately induce labor. Of course, like many old wives' tales, there's always that kernel of truth in it. One of the remedies if you are past your due date and still have not given birth yet, one of the old wives' tale is what got you into the mess can get you out of the mess. Right? So sex got you pregnant, and sex can potentially get you out of it as well. So again, that orgasm, those chemicals in the ejaculate can actually stimulate the onset of labor. What do you mean by ripen the cervix?
Like the knit, getting it ready to push the baby out. Not the cervix, but.
Okay, well, so again, it can cause the contractions, absolutely, that can do that as well. As far as the ejaculate, it's also the opposite as well. It isn't so much that it needs to ripen the cervix to let the baby out, but it does need to soften, for instance, the mucus plug that is providing protection to help to get the sperm through. So absolutely, so there's all these things necessary, but, and again, this is one of my all-time favorite things to say in this class, the vagina is a hostile environment. All right. Now, again, we all get to giggle when I say that. I like to giggle when I say that. But remember what we were talking about before. The female's reproductive tract is open to her abdominal pelvic cavity. And so as such, as we talked about, harmful things in the outside world, pathogens, bacteria, sexually transmitted diseases, if they get into the female reproductive tract, have the potential to get to any of the organs of the abdominal pelvic cavity. And so that is a very scary process or prospect. As such, we want to protect the female body as much as possible. And so the vaginal canal is very acidic. And that acidity helps to reduce any bacterial infections or any other types of pathogens from getting in there. And that's awesome and important, vital, in fact. The problem is that acidity slows the swimming motion of the sperm. Remember also, the sperm have to pass through the male urethra, which has urine in it, which, as we've talked about, is mostly acidic. So we have the acidity of the urethra, we have the acidity of the vaginal canal, and so part of the ejaculate needs to reduce that acidity so that the sperm can be more efficient in their swimming motion. And although I don't think it's here on the list, oops, nope, I don't have it here. Great question. Yep, we'll get to that in just a second. And it also needs to provide lubrication. Lubrication helps to facilitate copulation for intercourse. Lubrication helps to increase the enjoyment of copulation. And after all, if the goal is to propagate the species, you want that process that causes the propagation of the species to be something that you want the participants to participate in. So making it a pleasurable experience increases the likelihood of propagating the species. And so the lubrication can provide that as well. And yes, to answer your question, not to get ahead of ourselves, but yeah, the pre-ejaculate that is released from the male does those two things. It helps to provide lubrication for copulation, and it helps to cleanse the urethra of some of that urine to help to neutralize the acidity. And we'll actually see what glands make that in just a moment. But before we get to the pre-ejaculate glands, let's talk about the seminal glands, or what are also known as the seminal vesicles. These again are two paired glands. They are also located on the, oops, light doesn't show up well. Located on the posterior wall of the bladder. They form a majority of the ejaculate, making up close to 70% of the semen. And they produce a very thick, a very viscous, and a very alkaline kind of yellowish secretion. There are dozens of chemical components within there. I'm not going to rattle all of them off, but let's talk about some of the important components that we find in there. One of the things that is in the seminal fluid is fructose. Why might that be important? Yeah, provide that energy for ATP. Remember, that swimming process is going to require massive amounts of ATP. So having that, that sugar readily available is something that can definitely be helpful. Ascorbic acid, I've heard that before. What's the other name for ascorbic acid? Vitamin C. Ah, that's so that the sperm don't catch a cold, right? No, that's what we think of vitamin C for. But in this case, vitamin C plays an important role in the activation of the sperm, this turning on of the tail motors. The fluid of the seminal vesicle also has a coagulating enzyme in it. Why is that useful? Why do we want the ejaculate to coagulate? Yeah, not only to keep it together, but so it stays where you put it, right? The goal, again, remember the goal of this copulation is fertilization. To fertilize the female, to get sperm to the egg, you want to take all of the sperm, get them into the vaginal canal, and have them stay there, right? If you're not able to get the sperm to stay in the vaginal canal, then they can't get to the egg. So the advantage of a coagulating enzyme is that the ejaculate will stay where you put it. Right? Prostaglandins, another one of those alphabet soup words that you know you're going to have to spell at some point. Prostaglandins are those hormones we were talking about. That job is to stimulate the reverse peristalsis of the female reproductive tract. The female reproductive tract undergoes peristalsis, primarily outward. Again, this is open to the outside world. So anything that tries to get in, we want to push it out. Every 28 days, the female produces menses, which needs to be expulsed from the female reproductive tract, and all those things. So the peristalsis in the female's reproductive tract is primarily outward. As I mentioned, orgasm, but also the prostaglandins in the ejaculate cause a reverse peristalsis. And that reverse peristalsis helps to bring the sperm up the vaginal canal into the uterus and ultimately to the uterine tubes, which is where fertilization will actually take place. So again, we talked about how copulation can trigger and induce labor. This is the hormone that does that. All right. Like I said, there are literally dozens of other components that nourish and turn on the sperm and make the sperm physically capable of fertilizing the egg. If you were to just pluck a sperm out of the ductus deferens of a male, and in a petri dish, just plop it on top of an egg, it wouldn't be able to do anything. It needs to be activated. It needs to be stimulated. Its acrosomial cap needs to be made delicate and brittle so that it can release those enzymes. There's all these things that it needs to do so that it is capable of fertilizing the egg. And all those processes are condensed together into one fancy name. Becoming capable of fertilizing an egg is a process of capacitation, right? Capacitation is the activation of the head region. You decapitate someone when you take their head off, or capacitation is the activation of the head of the sperm. All right. Come on, there we go. Our second gland, the singular gland, the prostate gland, as we mentioned, surrounds the urethra at the base of the bladder. So it's a donut-shaped gland that encircles the urethra. Notice when you look at this illustration of it kind of shows all of these thin lines. It has a striped appearance. That's because these thin lines are dozens and dozens of small ducts that release their secretion into the urethra. So there isn't just one duct out of this prostate gland. There are dozens of them that feed out of it. And that's why it gets a very striped appearance to it. It produces a little less than 30% of the semen and produces a very milky white type of secretion. Again, dozens of different chemicals that are in this, and the majority of the chemicals that are in this are really about activating and capacitating that sperm, turning on the swimming motion and getting the head ready to fertilize that egg. Now, again, why most of you are probably familiar with the prostate, and I think I have a picture that shows this. There we go. Is prostatic cancer is a cancer that it can be very common in males. Basically, as males continue to live longer and longer lifespans, essentially every male, if he lives long enough, is pretty much 100% guaranteed to get prostate cancer. The good news about it is in most cases, it is very slow moving cancer. So when that 70 or 80-year-old is diagnosed with prostate cancer, they don't always treat it very aggressively, because in most cases, they are more likely to die of old age than they are to die of that prostate cancer. But there, I keep using that pesky word most. And that's because in some people, prostate cancer can be very aggressive, can metastasize and spread to other areas, and can be very dangerous and very deadly. So it is important, especially once a male usually reaching about age 50, to get, regularly have their prostate inspected. And how do they do that? There you go. As you notice, the rectum is right adjacent to the prostate. So the doctor puts on the glove, inserts the finger, and will palpate the surface of the prostate. Well, milking is something else. We'll talk about that in a second. We'll feel the surface of the prostate, looking for the overall size and also looking for irregularities on the surface that could be an indication of tumors. So usually they do that once a year. The term milking the prostate is that stroking of the prostate. For some males, there are sensory receptors here that are highly sensitive. And so when that prostate is manipulated rectally in this fashion, it can produce intense arousal as a result of that. So that there are some males that will like the palpation of their urethra and find it very arousing in nature. So yes, so there are some individuals who enjoy that experience. And as we'll see, it's not that different, homologous. There's a homologous process in females that we will talk about as well. Although females don't have prostates, let's be clear about that, sort of. All right. That is the prostate. What that leaves us with is this gland right here. And let's start with it here. Again, one of those terms that you know you're going to have to spell at some point or another, the bulbourethral gland, a big alphabet soup term. It is also known as the Cowper's gland, and I will accept the use of the term Cowper's gland. I want you to notice, sorry, I've got a tickle, hold on. I want you to notice something about the bulbourethral gland. It is a paired gland. We'll see that in the other picture in a minute. And the body of the gland itself, is located in the levator ani muscle. So the body of it is located in the levator ani muscle. But notice its duct enters into the spongy tissue of the penis, and it actually inserts and releases its secretions into the spongy or penile urethra. I think we have one other picture. Here we go. So notice here, this picture shows it nicely as well. Notice the gland itself is located in the urogenital diaphragm in the levator ani muscle, but its duct projects forward into the urethra. Now, notice when we talked about the prostatic gland and the seminal vesicles, they release their secretion upon ejaculation, helping to form the semen that is released via ejaculation. These bulbourethral glands, though, become active upon arousal. So upon arousal, they produce a very thick, very clear, very alkaline mucousy secretion that, as someone mentioned earlier, we commonly refer to as the pre-ejaculate. And as we just finished talking about, the advantage of this pre-ejaculate is that because it's thick, mucousy, and alkaline, it can help to neutralize the acidity of the urethra. It provides lubrication for copulation and can help to reduce the acidity of the vaginal canal. All right. Now, there's one more catch on this pre-ejaculate. Upon arousal, as we've talked about, these bulbourethral glands will become active, producing that pre-ejaculate that is released into the urethra. Exactly, that's where I'm going, Ashley. Arousal also causes the contraction of the ampullus. And when it produces the contraction of the ampullus, that will release some sperm into the pre-ejaculate. Now, the overall volume of sperm released from this is not necessarily as big, but also the overall volume of the pre-ejaculate is not as big. In fact, the concentration of sperm in the pre-ejaculate can actually be much, much higher than the percentage of sperm in the ejaculate itself, because the ejaculate itself has so many more fluids in it that the pre-ejaculate can actually have a higher concentration of sperm with it, which is one of the reasons why the withdrawal method is one of the worst methods for preventing pregnancy. Was it the pre-ejaculate that got you, Ashley, with your kid? Yep, exactly why. So that's exactly the problem with that. The pre-ejaculate can actually have a higher concentration of sperm in it than the actual ejaculate itself. All right. There. So there you go. Questions on that? All right, you put all this together, mix it in a pot, and you get our semen, or our ejaculate. A mixture of sperm and those accessory gland secretions, a milky white in appearance with a pH of about 7.3 to 7.7, so slightly basic, again, to neutralize that acidity. And again, remember, only 5 to 10%. I don't know why that says 10%. Closer to 5%, so let's fix that. It can be as high as 10, but in most cases, it's more closer to 5. So let's go with 5. I like 5 better. So about 2 to 5 milliliters of semen is released during an ejaculation, and only about 5% of that is sperm. Now, I know that doesn't sound like a lot, but remember how many sperm those males are making and how teeny tiny they are. In that typical ejaculation, a man can have somewhere between 20 and 150 million sperm per milliliter. All right, so again, the average ejaculate has 500 million sperm inside of it, which sounds like a massive amount, but that's actually needed. If a male has less than 25 million sperm per milliliter, he is considered to have a low sperm count. And if a male has less than 20%, pardon me, not 20%, if a male has less than 20 million sperm per milliliter, that male is considered infertile, incapable of getting a woman pregnant. 20 million sperm per milliliter, right? And yet incapable of fertilizing a female. So again, it's the ejaculate itself has a larger volume of sperm in it, but it also has a larger volume of secretions in it as well. Right? Again, the ejaculate, the semen, will have a total amount of 500 million sperm. But remember, that makes only up 5% of the ejaculate. The pre-ejaculate could have 30 million sperm in it, but it could be as much as 40% of the pre-ejaculate. So again, it's a higher concentration, but fewer overall sperm. So I wouldn't say that the pre-ejaculate makes it more likely to get pregnant than the ejaculate itself, but it is possible to get pregnant from the pre-ejaculate itself. So that's why just waiting till ejaculation and then withdrawing before ejaculation is not a way to avoid getting pregnant, because plenty of sperm are still going to be released prior to ejaculation. So that's the point of that. It isn't that it's more likely with the pre-ejaculate. It's just that pre-ejaculate contains sperm, so you can get pregnant just from pre-ejaculate. And again, like we talked about 60%, 60 to 70% are fully functional and viable in a well-rested male. But like I said, with continuous ejaculation, that number can drop below 50%. All righty. Questions on that? All right. With that, we are done with the male anatomy. We only have to do the male physiology, which is going to take us like 32 minutes. And then we're finally done with the Volkswagen Bug version, and then we get to move on to the Lexus. I would encourage you to get a head start. I'm not forcing you to think of this material by having an assignment due on Tuesday when we come back. And quite frankly, I'm not sure why I did not do that. you should have an assignment that you should be doing. So you're thinking about this material, but hopefully as you are in your 10th section of anatomy and physiology, you have learned that when you have large periods of time like this, you should use them to your advantage to study and get ahead. If you haven't figured that out by now, then there's not much I can do for you for the last 3 weeks of the class. So you don't have an assignment due on Tuesday when you come back, but please be responsible with your time. Study hard, be successful. No, there's no assignment due Tuesday. Again, your next assignment is due on, no, that is their schedule. Your next assignment is due on Thursday, the Labster. But again, there's no reason you can't get a head start on that if you want to do that, or just start studying this material and looking ahead. I don't have any scheduled extra credit opportunities that I'm aware of. Again, you still have, well, I guess if you haven't signed up for the Science Success Center, that does remind me one thing though. If you haven't signed up for the Science Success Center, it's too late to sign up for that now, but you still have the open lab opportunities. And I think I got an email from Jeff saying that he was going to have an open lab during break. Let me see if I can find that email. Where was that? So excellent. So this Friday, Jeff will be holding his open lab, the normal hours from 10:30 in the morning till 8:30 in the afternoon. So even though technically it is a school holiday and campus is closed, since he's not on campus, he will be doing the open lab. So that is still an opportunity for extra credit as well. So go, he's got some of the great models. Ask him to show you his penis and vagina, and I'm sure he's got plenty of those models that he would happily share with you. So because again, there's some good charts, there's some good models in the classroom, and those are the kind of things that you will be responsible for on the exam. All right. Excellent. I'm sorry, go ahead.
But you said that only 15 hours can help us, right? Like 15.
So, okay, you can only get 15 points of extra credit. Yeah. However, going and working and getting that material, putting 30 hours in may not get you 30 points of extra credit, but going to the open lab, listening to the material, having them go over the models for 30 hours, is that going to help you to be successful on the exam? Really, really help that way? Yes.
Absolutely. Yeah.
Let's try. I didn't think about it that way. Yes, he model of, so not his penis and vagina, but the models of the penis and the vagina. Yes. So thank you for clarifying that. All right. This is always an awkward section, but it's even more awkward when A, we're online, and B, all of your cameras are off. This is very awkward. So this is not fun. If I have to be uncomfortable, you guys should have to be on camera and be uncomfortable as well. All right. Have a good weekend. Have a good and safe Thanksgiving, and I will see you next week. Bye.